to all of you. I am seeing the chairperson as well as the.
He's already here.
Ravindran sir, good afternoon.
Yes, sir. Good afternoon, sir. Good afternoon.
Dr. Ravindran, good afternoon.
Yes, sir. Good afternoon.
Dr. Manjia. Shall I contact our director, sir?
He is joining, madam. Just 1 second.
Okay.
Yes. Yes, madam.
Good afternoon.
Good afternoon,
Can
sir.
we start?
We'll start, sir.
Yeah, Dr. Ravindran, very good afternoon. Welcome.
Very good
Good to
afternoon.
see you.
Thank you
Are
very
you
much.
muted?
No, sir.
Check
I'm
if you're
not
not
muted.
muted.
No, he's
Good
not.
afternoon, sir.
Okay, good. Okay, let's begin.
Still not,
the
we are
session.
able to hear you.
Yes. Should we start, sir? Good afternoon.
Okay, now it is fine. I think my side it was
Okay.
on the low volume. Good. So good and great to see you. We can start.
Yes. Good afternoon, everyone. And this we're now joined post-lunch session. This is the fourth session of the significant achievements to be presented by professors. And I warmly welcome our chairman today, who is chairing the session on the School of Basic Sciences. I'm very glad to welcome Dr. Ravindran, Dr. M.
Thank you very
Ravindran.
much, madam. Thank you.
He is a professor and head Department of Plant Biotechnology, Center for Plant Molecular Biology and Biotechnology at TNAU, Coimbatore. Sir, we warmly welcome you, sir, and we are very happy that you accepted our invitation and today you
Thank
have
you very much,
joined
madam.
us to guide all of us. And let me just surprise you, sir, because this is a school mode. All the professors are presenting the significant educational achievements made in their respective disciplines of the students who are going to get their degrees in this 60th convocation, which is going to be held on 11th February. So in the morning sessions, we had 3 sessions. First was for the School of Crop Improvement. Second was for the School of Crop Protection. And then we had the School of Natural Resource Management. So post-lunch, we are beginning with the School of Basic Sciences. So School of Basic Sciences, as you're all aware that we have 3 disciplines in the School of Basic Sciences: biochemistry, molecular biology and biotechnology, and plant physiology. So in total, there are 21 theses work, which will be presented by all the 3 professors of these respective disciplines. And there are 17 MSc students and 4 PhD students who will be getting their degrees during this convocation. So, and I also welcome our director, who has been guiding us all through, and also the convener of this session, who is continuously conducting all the sessions since morning, Dr. S.P. Datta, who is professor in soil science and agriculture chemistry. So he will be conducting the session for further. Before that, I would like to invite our director to formally introduce our chairperson of today's presentation.
Thank you, Dr. Rasmi, and a very good afternoon to each one of you. It's my proud privilege to introduce to this August gathering the chairperson of this session, Professor M. Ravindran. He is a renowned molecular biologist, and he did his PhD in plant biotechnology from TNAU. and carried out his postdoctoral research at the International Rice Research Institute in the area of genomics and bioinformatics. He has been deeply engaged into teaching and research, and he has offered more than 30 courses to UG and PG students, and mentored and sponsored postdoctoral fellows, 5 PhD students he has guided, and 25 master's students. Several of his master's students have secured national level fellowship of different kinds, and international fellowship as well. And he has a large number of projects from the various funding agencies, including DVT, DST. He is also a resource person for DVT. In most projects, he is in the task force. And more importantly, he is a very involved researcher. And one of the classical example of his, association out of box thinking and out of discipline, which I impressed the most me, was that there is a Ganga Hospital in Coimbatore, and that's a hospital of orthopedics. And there, I think Dr. Chandrasekhar, Dr. Sekran, he is a renowned surgeon, and looks at the disc degeneration in people. He had a lot of phenotypic data on the disc degeneration from people coming from different parts of the country. This is a very renowned hospital. And then Dr. Ravindran, in association with Dr. Shekhar, they try to look at the molecular basis of disc degeneration, how it happens. So you can see someone who is working in agriculture field and collaborating in the medical sciences carried out an association mapping by genotyping these individuals and phenotyping data from disc degeneration, and was able to really establish a relationship as to people coming from which origin have got more sensitivity and are prone to degeneration, and so on. So that was a very interesting study. And he has been associated with almost 150 research papers published, and has been associated with development of varieties Co43, sub-1 version, and Co52. And we had a great collaboration, and still we have on one project on, Nagina 22 project that was on tilling, through which he and his colleague, Dr. Robin, and they came out with the herbicide-tolerant mutant for the first time. So he is a recipient of several honors and awards from Tamil Nadu Scientist Award, Tamil Nadu State Council of Science and Technology, and Best Researcher Award of TNAU. His commitment to science is remarkable, something to really emulate for anyone to see, and how involved one can be in research. Most of the times you will find him in the lab, even holidays, or working days, late hours. Teaching involvement is great. He has got excellent capabilities in bioinformatics along with molecular biology. So it's my proud privilege and pleasure to welcome Professor Ravindran to share this session on basic sciences. Welcome, Ravindran. Thank you very much.
Thank you. Thank you very much, sir, for your introduction.
Yeah. Thank you, sir. Thank you very much. Now we are at 4th school, that is School of Basic Sciences. And this school is also very important in the sense that basic research is precursor of applied research. And once I remember my teacher actually at my MSc days, he told that applied and basic sciences, these are just like our two legs. We cannot work with, as we cannot work with one leg, science also cannot progress without either of them. So we have 3 disciplines, as Dean Madam already made mention: biochemistry, molecular biology and biotechnology, and plant physiology. So with your kind permission, sir, Chairman sir, may we take up presentation of molecular biology and biotechnology?
Yes, sir, sure. We can.
Okay. Okay, thank you.
Yeah, and before that, Dr. Datta, I also see Dr. Lodha has joined, one of the greatest teacher that we had in the biochemistry. And the way he used to conduct his practicals and the capacity building in the area of molecular biology, particularly techniques, used to be remarkable. People remember him always. Welcome you, Dr. Saab, and I'm so glad to see you. Thank you so much.
Thank you very much, Dr. Singh. Thank you.
Good afternoon, sir.
Honorable Chairman of the session, Director IARI, Dean PG School, Convener of this session, all the dignitaries, faculties, and students. Let me present significant research achievements of the Division of Molecular Biology and Biotechnology. Total 22 courses have been offered in 2 semesters by 30 faculty members. At present, total 64 students, 17 MSc and 47 PhD students are pursuing their study in the division. This year, 9 students, 7 MSc and 2 PhD students are receiving degree. Research works are categorized into 3 broad theme areas: abiotic stress management, biotic stress management, and basic study. Research works of 4 MSc and 1 PhD degree recipients were aimed to improve abiotic stress tolerance of crops. Salinity stress is a serious concern for rice cultivation in India. Ms. Rekha Mahato studied salt tolerance in sensitive rice genotype IR29 after exogenous application of allantoin, a metabolite of urate pathway, under the guidance of Dr. Tapan Mandal. Increased root and shoot length and higher fresh and dry weight of root and shoot were recorded in seedlings treated with both NaCl and allantoin. Similarly, proline and allantoin contents were higher in seedlings treated with both NaCl and allantoin. Relative expression of hormonal biosynthetic genes was studied. Upregulation of genes involved in biosynthesis of auxin, gibberellins, ABA, and brasinosteroid was observed under both NaCl and allantoin treatment. It was concluded that allantoin acts as a signaling molecule and activates endogenous pathways involved in salinity tolerance. Multiprotein bridging factor 1, or MBF1, is reported to be involved in abiotic stress tolerance. Mr. Muhammad Asik did phenotypic characterization of heat stress responses in 2 contrasting wheat genotypes and in silico analysis of MBF1 gene in wheat under the guidance of Dr. Sarmishta Barthakar. Phenotypic analysis revealed that heat stress not primed susceptible cultivar HD2967 showed better basal thermotolerance than that of resistant cultivar Raj 3765. However, both the cultivars, when primed for heat stress, showed acquired thermotolerance. In silico analysis revealed that MBF1 homologous genes present in 7th chromosome of A, B, and D genomes are upregulated under both biotic and abiotic stress. MBF1 protein is predominantly localized in cytoplasm and nucleus. MicroRNAs regulate MBF1 gene expression post-transcriptionally either by transcript cleavage or by translational inhibition. Phylogenetic analysis grouped 82 MBF1 protein coding genes from 19 selected plant species into 15 major clades. It is concluded that the structural information would help to identify MBF1 homologs for functional analysis. Calmodulin, or CAM, is a well-characterized receptor for calcium, and it participates in signal transduction pathway of biotic and abiotic stresses. Ms. Seta characterized CAM gene from pearl millet, which is highly tolerant to various abiotic stresses, and studied the role of the protein in heat stress tolerance under the guidance of Dr. Jasdeep Padaria. The pearl millet CAM gene was introduced into binary vector, and the recombinant vector was used for transformation of tobacco leaf explants. Putative transformants were screened by PCR using gene-specific primers. Potted transgenic lines were exposed to heat stress. Relative water content, chlorophyll content, and membrane stability index increased in transgenic lines compared to that of control plants. Higher expression of the CAM gene was observed during heat stress. The study showed that the pearl millet CAM gene is involved in heat stress tolerance. Salinity stress is one of the major factors affecting wheat production. Khachia wheat is known for its tolerance to salinity. The underlying mechanisms of salt tolerance in Khachia are not well understood. Ms. Ankita Chinche carried out molecular and biochemical analysis of salt tolerance in real mapping population of Kharchia 65 and HD 2851 under the guidance of Dr. Kanika. Based on cluster analysis of physiobiochemical, root structural, and growth parameters, it was found that reals in real 5 with salt tolerant Kharchia 65 and real 13 and 14 were grouped with salinity susceptible HD 2851. Based on these results, tolerant real 5 and susceptible real 13 were selected for gene expression analysis. Salinity-related genes encoding sodium hydrogen ion antiporter and vacuolar pyrophosphatase similar to AVP1 were upregulated in leaves and roots of both the real 5 and real 13. However, higher upregulation was observed in real 5 as compared to real 13. Mr. Lal Bahadur Singh identified and characterized drought stress responsive microRNAs in chickpea at terminal flowering stage, with the aim of better understanding of regulation of gene expression in chickpea in response to drought stress under the guidance of Dr. Pradeep Jain. High throughput sequencing of small RNA libraries followed by sequence analysis revealed presence of 224 microRNAs belonging to 51 conserved families. The position of these microRNAs had been marked against their genomic coordinates. Based on expression analysis, novel microRNAs were identified that are upregulated in tolerant genotype during drought stress. An inverse correlation between the expression of selected novel microRNAs and their target genes in root tissues of both tolerant and susceptible genotypes was observed, suggesting that these microRNAs might have a role in drought stress response in chickpea. Research study of one MSc degree recipient was aimed to improve plant biotic stress tolerance. Bacillus thuringiensis, or BT, is widely used in agriculture for the control of insect pest. Mr. Asfaq Mujawar screened BT isolates collected from different agroclimatic zones of India for the presence of genes encoding vegetative insecticidal protein 3 type under the guidance of Dr. Sarabjit Kaur. The BT isolates were screened with different set of primers. Complete ORF of VIP3 type gene was PCR amplified using primer set 1 from a number of native BT isolates. The ORF was cloned onto PGM vector and sequenced. Sequence analysis revealed that the amplified genes had both nucleotide and amino acid substitutions compared to the already reported VIP3A type genes. Homology modeling of one VIP3A type protein and scavenger-like receptor C showed hydrogen bonding and other non-covalent bonding between the two proteins. It was concluded that these VIP3A type genes could serve as valuable source for deployment in transgenics for insect pest resistance. 2 MSc students and 1 PhD student carried out basic research work. Hydrotropism plays important role in drought avoidance. MEJ1 is the first gene characterized for hydrotropism in Arabidopsis. Towards characterization of MEJ1 homologs in wheat, Mr. Akash Maiti studied subcellular localization of MEJ1 homologs using green fluorescent protein or GFP as a reporter system under the guidance of Dr. Monica Dalal. 4 MEJ1 homologs located on 4 different chromosomes were amplified by RT-PCR and cloned to develop GFP MEJ1 fusion constructs. The constructs were confirmed by restriction analysis and sequencing. Confirmed constructs were transformed into agrobacterium and used for transient expression in onion peels. Subcellular localization of the fusion protein was analyzed using confocal microscope. MEJ1 protein is predominantly localized in cytoplasm and also surrounding the nucleus. This study provides basic information on subcellular localization of wheat MEJ1 homologs. Ms. Anindita Borua's research study was aimed at identification of genome-wide distribution and cloning of the cytokinin oxidase dehydrogenase 5 or CKX5 gene in Brassica janssia under the guidance of Dr. Naveen Gupta. Partial gene of Brassica was PCR amplified using primer designed from conserved region of CKX5 gene sequences of different Brassica species. The gene fragment was cloned and sequenced. The 2 paralogs of CKX5 present in Baruna genome were identified on chromosomal scaffolds A7 and B7. Genomic structures of both the paralogs were analyzed. Phylogenetic tree shows that the A7 paralog is closely grouped with CKX5 gene of Brassica rapa and could be orthologs. The B7 paralog is individually placed in a separate clade and could be descended from another progenitor, Brassica nigra. Photoperiod sensitivity is a critical constraint limiting PGNP productivity. Mr. Krishore Tribhuvan studied to understand the molecular mechanisms underlying photoperiod response in PGNP. Resequencing of photoperiod sensitive and insensitive genotypes, followed by variant analysis, identified a high number of SNPs and indels in the genomes of these two genotypes. Genes of plant circadian rhythm pathway containing SNPs and indels were identified. 7 candidate genes for photoperiod sensitivity in PGNP were identified through expression analysis of SNPs and indel containing genes. Based on quantitative expression analysis of different genes involved in flowering were identified that showed upregulation in short and long day conditions in the photoperiod sensitive and insensitive genotypes. It was concluded that flowering induction appears to be regulated by both photoperiod dependent and photoperiod independent pathways working simultaneously. A number of publications in different NAS rating journals have so far been published by the degree recipient students. MBB faculty published 86 research papers in the year 2020. Happy to inform you that majority of degree recipient students are well placed. 3 MSc degree recipients are pursuing PhD at IARI. Both the PhD degree recipients are engaged in government jobs. Throughout the year, all the teaching and other activities were conducted online. These are some snapshots of different virtual activities conducted by the division. With humility, I thank all for your guidance, support, and contribution. Thank you.
Thank you very much, Professor Pramanik Patanayak, for your nice presentation. And with the permission of chair, we will move to next presentation, plant physiology. Professor Madan Pal Singh will make presentation.
Everybody, Honorable Chairman and resource persons of this session, I am Dr. Madan Pal Singh, Professor of Plant Physiology, presenting the significant postgraduate research achievements from the discipline of plant physiology. Thesis submitted in plant physiology during 2021 represent two major theme areas. The first one is plant nutrition and second is hepatic stress tolerance. In plant nutrition, 3 MSc and 1 PhD students submitted their thesis. Sinto worked on interactive effects of elevated CO2 and nitrogen fertilization on yield and nitrogen response of bread wheat under the guidance of Dr. Lakshmi. The hypothesis of this study was that supplying of additional nitrogen during reproductive phase might improve the grain yield and quality under elevated CO2 environment in wheat. The study was conducted in 2 seasons. I am presenting here the data of 2021. These are the details of the nitrogen supply at different stages, and these are the photo open top chambers which have the two treatment, the AC ambient CO2 and EC elevated CO2. These graphs shows the utilization and remobilized efficiency under EC in wheat, and NUT at the harvest was significantly lower in EC and nitrogen treated plants, and highest was found under EC with low nitrogen treatment. Similarly, NRE increased in AC grown plants and highest was observed in lower nitrogen treatment under AC. Post-synthesis nitrogen uptake increased under elevated CO2 and highest was found with ECO2 and high nitrogen treatment. Normally, grain protein content declines under elevated CO2 environment, but in this study it was found that there was no reduction in the grain protein under elevated CO2, and with additional supply of the nitrogen at the reproductive stage, there was significantly higher protein content compared to the ambient CO2. These graphs shows the 1000 grain weight and grain yield per plant. In general, the elevated CO2 exposure results into enhanced 1000 grain weight and the grain yield. But here in this study, there was a reduction in both these parameters, and it was observed that it may be occurred due to the reduction in grain size under elevated CO2 environment. These bar graph shows the effect of elevated CO2 and nitrogen fertilization on grain size of the bread wheat. It was found that both the genotypes, there was increase in grain length under elevated CO2 than in the ambient CO2. While the BTS genotype, the grain width was found to be decreased under the elevated CO2 and HRN treatment. As reported earlier, that EC treatment increased the grain length and decreased the grain width under HRN treatment in this genotype. This can be clearly seen from the photograph in the HRN treatment. So it was due to the higher expression of the related genes to grain length and the grain width, and which are the basically negative regulator under the EC and HRN treatment, whereas the expression of these genes was reduced under the low end supply. Nidhi Chaturvedi worked with Dr. Dandapani on jivas of nutrient use efficiency and regulation by nitrogen stress. 300 diverse rice genotypes were grown under control environment, and grain nutrient data was used for jivas and QTL analysis. The slide shown the grain macronutrients in diverse rice genotypes under the controlled nitrogen stress conditions. In general, except the manganese, all the nutrients decrease under nitrogen stress conditions. As shown earlier, the level of grain nutrients decrease under nitrogen stress, but some genotypes are found to have highest iron and zinc under stress and selected as donors. In the Jivas study, 51 QTLs were found under normal nitrogen conditions, and among them, the 40 were the novel and 11 were the already reported. Similarly, under nitrogen stress, 39 QTLs were found, and among them, 29 were the novels and 13 were earlier reported. This slide shows the co-localized QTLs for rice grains. It was found under normal nitrogen treatment. The 4 co-localized QTLs were found, and the 3 were found under stress conditions. And the study concluded that these co-localized QTLs can be used further for improving the nutrient in grains. Also, the annotation of the novel QTLs can lead to finding the putative genes for the nutrient uptake mechanisms. Also, the contrasting donors can be utilized to develop the high macronutrient accumulating genotypes. This is the work of Anime Reddy with Dr. Sudhir Kumar. He worked on the grain quality in wheat under different nitrogen conditions, and the study was similar to the earlier one in rice. Here they have taken 190 tutorials and their parents grown under the control environment, and on maturity the grains were analyzed for the nutrient levels, and the data of the nutrients was used for jivas and QTL analysis. Network plot correlation showed association between different elements in grains. Dark blue line showed a strong and positive association, and the light blue indicates peak association. In case of red light, both elements are not associated or have negative association. Multi-trait genotype endotype distance index analysis led to identification of 10 superior genotypes performed better under the nitrogen stress and accumulated higher elements in grains. Then, using this data, the jivas was conducted and the QTLs were analyzed, and there was a robust QTL found for the iron with LOD score of value 13.42 with a PV percent value of 10% on chromosome 1. Further jivas is going on, and more information on QTLs will be available later as the nutrient analysis were delayed due to some reasons. Parvat did PhD with Dr. Ishwar Singh, and she worked on characterization of physiological molecular response on maize associated with adaptations to nitrogen stress. So in this study, they have taken 40 inbred lines which were raised in the normal and deficient nitrogen in soil and screened on the basis of plant biomass and economic yield. On the basis of the morphological and physiological traits, the cluster analysis of the genotypes was done, and they were grouped into the two clusters, cluster 1 and cluster 2. This is the dendrogram of the most contrasting 15 maize inbred lines. The C cluster 1 represents the nitrogen efficient and cluster 2 represents the inefficient lines, and this is the nitrogen efficiency percentage was calculated for both the clusters. From earlier study, 2 contrasting genotypes were selected and they were grown hydroponically. After 21 days, the tissues from the shoot and root were collected and transcript analysis was done. These are the details of the genes analyzed, and this is the growth performance of 21-day seedlings of the contrasting genotype under the normal and deficient N. This Venn diagram represents the number of genes upregulated and downregulated under normal and nitrogen deficient conditions in contrasting genotypes, and the circles plot depict the distribution of the differentially expressed genes on 10 chromosomes of maize. This is the heat map depicting the top differentially expressed genes with p-value of the less than 0.05. These are the down-regulated genes, and these are the top 20 upregulated genes. The green line indicates the down-regulation, while the red line indicates the upregulation. This slide shows the expression level of the selected nitrogen-responsive genes using the QRT-PCR and NGS data in response to the nitrogen stress. The y-axis here represents the log to fold changes in the expression level of the genes, and these results validated the expression of nitrogen responsive gene as similar to the NGS data. And this study confirms that overexpressed genes and nitrogen efficient genotypes can be used in future research programs. Under theme 2 on abatic stress tolerance, 2 MSc students submitted thesis last year. Jyoti worked on hormone regulation of stagnant trait under heat stress and wheat under the guidance of Dr. Pramod Kumar. The study involved 2 major activities, that is identification of contrasting genotype for stagnant trait and hormone regulation of the stagnant trait under the late planting conditions. This photograph indicates the performance of the heat genotypes under the normal late planting conditions. In the first activity of this study, 32 wheat genotypes were screened under late planting conditions on the basis of the spread and pigment degradation, And using this data, the quantitative analysis was done of all the genotypes, and among them, 2 sensitive and tolerant contrasting genotypes were selected. In the second activity, the contrasted genotypes were grown in the same environment with 5 subtreatments of the foliar application of cytokinins and inhibitors, and it was found that heat stress enhanced the leaf senescence, reduced leaf area per plant and NDVI, and non-stick green type genotype was found more sensitive, but the application of the 2 cytokinins delayed the senescence, maintaining green leaf area and NDVI for longer duration of the time. Similarly, rate of photosynthesis and chlorophyll concentration decreased with the heat stress, and the foliar application of the cytokinins improved the rate of photosynthesis as well as total chlorophyll concentrations. This slide shows that application of the cytokinins improved the heat tolerance index and lowered the heat susceptibility index, and the response was better in tolerant or stick green type. Finally, this study concluded that under late planting conditions, the wheat gain yield can be improved by enhancing the stick green trait by foliar application of the cytokinins due to increase of photosynthesis, concentration of pigment, and improvement in the membrane stability and the antioxidant system. Gopadev worked with Dr. Vijaypal on physiological characterization of mutants for on length in wheat, and they were taken the 6 mutant line to the popular variety 3086 and grown under the normal and heat stress conditions. And these are the photographs of the wild type and also the mutants with the variable on length. This is the on temperature of the main year at the anthesis and post anthesis stage. The on temperature was measured using the thermal imaging, and it was found that all the 4 mutant lines have the higher temperature compared to the wild type lines, and this high temperature of the ons was associated with the shortness of the ons. Similarly, the tissue temperature of the main year was recorded at both the stages, anthesis and post anthesis stage, and the data shows that in comparison to the control plants, 3 mutant lines had higher main year temperature at anthesis stage, while at the post anthesis stage, the year temperature of 4 mutant lines was lower than control, and it might be associated with the better gain yield of these mutant lines. This slide shows the total gain yield per plant decrease in all the mutant lines, and heat susceptibility index, which was calculated on the basis of gain yield per plant. 4 of the mutant lines were found to be tolerant to the heat stress, while 2 of them were found to be heat susceptible. So this study finally concludes that ear or on tissue temperature is a very important trait and is dependent on the on size under both the environments, and it can be further exploited. Pratik received the IRA MSc Merit Medal in last year, and now he is pursuing the PhD at Danforth Plant Science Center, Washington University, USA. Center got the Young Researcher Award, and the Nidhi Chaturvedi got the Best Poster Award, and also jointly they got the Science Quiz Competence Award at the National Conference of Plant Physiology 2021. Before I close my presentation, I would like to thank our Director IRI, Joint Director Education and Dean, Associate Dean, our Head of the Department and School Coordinator, all the faculty members of the Plant Physiology, chairpersons and students who submitted their thesis, PG school staff, administration, technical supporting staff of the division, and finally, I thank one and all for your patience listening.
Thank you, Professor Madhurpan Singh, for very nice and excellent presentation. Now we will move to the last presentation of this school. Professor Anil Dahuja will make presentation on behalf of biochemistry.
Honorable Chairperson of this session, Director IRI, Dean and Joint Director Education, and other esteemed members of the IRI fraternity, I am going to present the significant educational achievements of the discipline of biochemistry made during the year 2021. Before actually moving on to the research findings and academic accomplishments of the students and faculty members, I would like to give you a brief overview of the division. We have a total of 12 faculty members and 36 PG students at present. We are running 18 courses in the trimester system and have started 16 courses in the recently introduced semester system. This year, 5 MSc and 1 PhD student will be receiving their hard-earned degree in the discipline of biochemistry. The research work carried out by the students can broadly be categorized into 3 theme areas: non-destructive nutrient profiling of food crops, the second one death-spring factors controlling quality, bioavailability and functionality of nutrients, and the third one enhancing the nutritional and keeping quality of the food products. In the first theme area, 1 PhD student did his thesis work, while 3 MSc students worked in the second theme area and 2 MSc students worked in the third theme area. The first PhD student, Maharishi Tomar, he worked under the guidance of Dr. Archana Sachdev, and their objective of their study was the evaluation and identification of the pearl millet genotypes based on the nutrition quality, biochemical diversity, and physiochemical parameters, and these are the objectives of their study. The major goal of this study was to develop eco-friendly analytical methods for high-throughput screening of the pearl millet genotypes. And to achieve this goal, so they developed NRIS-based model for nutritional profiling of the pearl millet genotypes. They screened the 87 diverse pearl millet genotypes, including land races and the commercial varieties, and then they did the profiling of the different nutritional parameters like carbohydrates, proteins, lipids, anti-nutrition factor, phenol, and the ash. And after doing all these analysis, they divided the 87 genotypes into the 7 clusters. Then they studied the correlation coefficient between the different nutritional parameters, and they also developed the heat maps for this purpose. And this work was published in the Journal of Food Composition and Analysis, having NAS rating 10.55. They also developed the near infrared spectroscopy based prediction model. So based on the approach which is given here, and this is the first report on the MPLS regression model for the rapid quantitative determination of the starch, resistant starch, amylose, protein, oil, total dietary fiber, phenols, total soluble sugars, and the phytic acid. And this work again has been published in a very good journal, the LWT Food Science and the Technology, having NAS rating 10.95. One MSc student, Mr. Tamil Selvan, worked under the guidance of Dr. Venuta for deciphering the influence of food matrix state of composite flour on nutrient quality and bioavailability, and these are the objectives of his study. The major goal of their study was to provide sustainable solution to the menace of protein and micronutrient malnutrition, and for this purpose they developed cost-effective pearl millet chickpea nutri-mixes with dough quality comparable to the wheat. And the pearl millet, they used both the sprouted grains as well as the unsprouted grains. So this was the composition of the nutri-mixes prepared by them. So as you can see here, the 6 nutri-mixes, they were developed. In the first 3 types of the nutri-mixes which they developed, they used the pearl millet flour directly, while in this 4th, 5th and the 6th they have used the sprouted pearl millet flour, and they used 3 different concentrations of the chickpea flour ranging from the 20% to 40%. So in the NM1 you can see 20%, NM2 30%, and NM3 it is 40%, and similarly in case of NMN4, 5, 6 the chickpea flour concentration is 20, 30 and 40%. So they carried out the nutrition composition analysis, and they measured the levels of the total crude and the soluble protein content. And as you can see here, as the concentration of the chickpea flour it is increased, so there is more increase in the content of the total soluble protein as well as the crude protein. They also determined the essential amino acid score of these nutri-mixes, and as you can see here that nutri-mix 3 and the nutri-mix 6, which are rich in the chickpea flour, so they have the very balanced amino acid score. They also carried out the dough quality analysis using the phrenograph test, and as you can see here in the unsprouted MN3 showed higher water absorption, dough consistency, and a lower dough development time as compared to the all other nutri-mixes. The microstructure of the gluten network and the starch protein interaction in dough matrix, it was also studied, and as you can see here, they also studied the in vitro starch digestibility and inherent glycemic potential. So there was increase in the in vitro starch digestibility and inherent glycemic potential in case of sprouted mixes, while in case of the unsprouted one, so increased SDS and the decreased RDS values they were observed. Another student, Ms. Debarati Mandal, who presented her research finding a day before yesterday for the IRI Merit Medal presentations, she worked on the starch-lipid interaction and its influence on the inherent glycemic potential under the guidance of Dr. Vedakrishnan, and these were the objectives of their study. The major goal of their study was to develop a cost-effective household strategy for reducing the glycemic profile of cereals for diabetic people, and for this purpose they studied the effect of exogenous addition of the cooking fats on the modulation of the inherent glycemic potential of the pearl millet and the rice. And they studied following these factors: starch dynamics, microstructure, matrix composition, the starch-lipid interactions, molecular patterns. All these factors they were evaluated to study the glycemic potential. They used 2 cereal sources, pearl millet and rice, for their study, and 5 cooking fats, the ghee, coconut oil, sunflower oil, mustard oils, and the til oil. And what they have found is the maximum complexation occurred in case of pearl millet, mustard oil, and the rice mustard oil, and the lowest iodine spectra was also observed in case of the mustard oil also. After the addition of the mustard oil, what they found was that there is a high level of the long range and the shorter range crystalline structure of the starch, and the compact structure ultimately hindered the enzyme accessibility and the glucose release. They also studied the starch lipid interactions using the confocal microscopy, using different types of the dyes, and the superior interactions they were observed when mustard oil it was added in the pearl millet and the rice. Another student, Mr. Kankan Pandit, worked on the genome-wide identification, cloning, and characterization of the genes coding for the lipase from the pearl millet and their physicochemical characterization for grain quality under the guidance of Dr. Ranjit Ranjan Kumar, and the objectives of the study are given below. The major aim of their study was to improve the shelf life of the pearl millet flour, and for this purpose they studied the gene expression analysis of the lipase enzyme, and they carried out the antioxidant profiling of the hybrids, composite, and land races of pearl millet having contrasting keeping quality of the flour. So these were the 4 varieties which were used by them, and this is the scheme of the things which they followed for the cloning and the characterization of the genes encoding for the lipase. The putative genes for the lipase and the triacylglycerol-lipase, they were cloned and they were submitted to the GenBank, and the accession numbers they were also obtained. They also carried out the semi-quantitative RT-PCR of the putative lipase and the triacylglycerol-lipase in diverse genotypes of pearl millet, and what they found was that there was a significant variations in the accumulation of the transcripts of putative lipase and triacylglycerol-lipase in land races, hybrids, and composites of pearl millet, and the abundance of the transcript it was more in the milli-ripe stage as compared to the milky ripe. They also carried out the biochemical characterization of pearl millet genotypes. They estimated the total antioxidant capacity, superoxide dismutase activity, catalase activity at different stages of the development, and also the free amino acid and total phenolic content and tannic content they were determined, and they correlated it with the activity of the lipase enzyme. Another student, Ms. Arpita, worked on understanding the effect of probiotic fermentation and germination on the protein digestibility, bioavailability of isoflavones, and the mineral content in soya milk. She worked under the guidance of Dr. Anil Dahuja, and these are the objectives given. Major aim of the study was to enhance the consumer acceptability of the protein-rich food sources, and they wanted to improve the nutrition and sensory properties of the soya milk through combined treatment of germination and the probiotic fermentation. So these are the 2 different types of the soybean genotypes used by them. One was the food grade, which contains high amounts of the protein, and another one the oil seal type of the soybean, which is rich in the oil, and these are the 5 strains of the Veziella which were used for the probiotic fermentation. So this is the strategy which was used by them. So they determined the beta glucosidase activity, phytase activity, and also studied the isoflavone bioconversion, because the bioavailability of the conjugated isoflavone it is less. So during the fermentation process there is a possibility that these isoflavones they can be converted into the free forms and their bioavailability can be enhanced. So they also determined the phytic acid content, cell viability, titrable acidity, and sensory evaluation it was also done of the probiotic drink which was developed. They determined the pH and the titrable acidity in different probiotic fermentation which was carried out by 5 different strains of the Veziella, and what they found was the pH was maintained at 5.6 to 4.6 in food grade soy milk fermentation and 5.74 to 4.6 in low KTI soy milk fermentation, and further the lactic acid also increased from 0.32% to 0.62% in soya milk from low KTI and from 0.17% to 0.32% in fermented soya milk from food grade. Maximum increase was seen in case of the Veziella sebaria SB. Further, they carried out the cluster analysis for the selection of the strain for the mixed culture approach, and the mixed culture approach was found to be more reliable than the single culture method of fermentation. And also they found that soy milk prepared from the germinated seedling has increased functional properties, yet overall acceptability of the product was low. The soy milk prepared from the fermentation of the food grade variety, which has shown higher acceptance after the addition of the sugar. Also, they observed 9.8-fold decrease in the phytic acid during the fermentation process, which further resulted in the increase in the levels of the zinc and the iron content in the soya milk. They also studied the isoflavone bioconversion during the fermentation process, and what they found was there is an increase in the A glycan forms observed both in the food grade as well as low KTI soy milk, and with maximum A glycan formation in the low KTI variety at 48 hours fermentation process, and the HPLC method was used for studying the composition of the isoflavone. Another student, Ms. Sushmita J, worked under the guidance of Dr. Arna Tyagi, and she worked on arresting the acidity of the rice bran by regulation of the lipase and the lipoxygenase enzyme, and these are the objectives of her study. For their study, they used following 4 treatments: the oven heating at 100 degrees centigrade, autoclaving at 120 degrees centigrade, infrared radiation treatment at 100 degrees centigrade, low temperature treatment at minus 80 degrees centigrade. And what they found was after these treatments, there was increase in the IL content in the rice bran. All these treatments resulted in the reduction of the lipase and the lipoxygenase activity, but the maximum decrease it was observed after the autoclave treatment. This is how the other parameters like iodine value, the sponification value, and the peroxide value, and the free fatty acid content, which were altered after these treatments. And what they have found is the autoclave resulted in the maximum decrease in the peroxide value, while the infrared treatment resulted in the maximum decrease in the sponification value. So overall, 28 research papers they were published by the faculty and the students, and I am happy to share that out of these 28, the 16 research papers they were having NAS rating more than 10, while 3 research papers had NAS rating between 8 and 10, and 8 research papers they were having NAS rating between 6 and 8, and only 1 paper was there which was having NAS rating less than 6. Also, 2 training programs they were conducted by Division of Biochemistry. The one was on journey of pearl millet from land to lab to plate, which was organized under the auspices of Niche Area of Excellence project, while another training on biomolecules, bioresources and biotechnology, it was organized under the auspices of the Society of Plant Biochemistry and Biotechnology. So these are some of the awards which were won by the faculty members. To name a few, the Incredible Scientist of India award, it was given to the Dr. Suresh Kumar, while the Fulbright Narrow Postdoctoral Fellowship award, it was given to Dr. Veda Krishnan. The students of the Division of Biochemistry were also decorated with certain awards. Ms. Debarati Mandal, who presented her thesis work for the IRI Merit Medal presentation, so she won 2 Best Poster awards, and then Simardeep Kaur has been selected as the principal candidate for the Nehru Doctor Fulbright Fellowship. So with this, I offer my sincere acknowledgements to Director IRI, Dean and Associate Dean, PG School IRI, Joint Director Research IRI, Head Division of Biochemistry, Biochemistry Faculty, students of the Division of Biochemistry. Thank you all for your attention.
Yeah, thank you very much, Professor Onil Dahuja, for your excellent presentation. Now, Chairman Sir, with this, we could cover all the presentation of School of Basic Sciences.
Yes, sir.
So now I request you, sir, for your kind remarks and comments.
So, very good evening, everyone, and thank you very much for the opportunity provided. Most respected Director IRI, Dr. A.K. Singh, and Joint Director Academics of IRI, Joint Director Research, Division Heads, fellow scientists, student friends, and ladies and gentlemen. Good evening to everyone. So indeed, it's an honor for me to witness the academic proceedings of School of Basic Sciences on the eve of your 60th convocation. I take this opportunity to congratulate everyone and also to wish everyone on the occasion of your 60th convocation, which is going to be held on the 11th of February. And I could see from the presentation that so all the 21 research thesis research activities or experiments were planned very well meticulously by both the guides and the students, and the students were able to execute the plan so without any deviation, and they were able to come out with improved knowledge on the respective areas. So before getting into the individual topics, one general observation is that so they have come out with very good track record of publication. From molecular biology and biotechnology, there were around 60 papers, and from biochemistry, there were around 20 papers of high impact. But I'm seeing So the number of PhD is a little bit lesser, only 4 PhDs out of 3 major disciplines. And both the administration and the faculty will consider this and try to get in more number of PhDs in the coming years. So coming to the individual research wise, and most of the research activities, so they were very focused and problem solving research, I can say. Like, so many of the thesis research have potential for translation into products. So say, for example, the soy milk study, and then alteration of glycemic potential of your carbohydrates in combination with fatty acids, and then manipulation of your photoperiodism in PGNP, and the manipulation of this one stay green trait, and then improving salinity tolerance. Many of them are having definite potential for translation into product technology, at least. So coming to the titles one by one, in molecular biology and biotechnology, under MSc, there was 2 PhDs. So both of them are well-conceived ideas. So one is a typical classical molecular biology towards identifying drought-responsive miRNAs in chickpea, and another one is understanding the molecular mechanism of flowering in response to photoperiodism in PGNP. So both of the studies, so they were carried out in a well-planned manner. So in the first study, miRNAs in chickpea, so they were able to come out with the 3 miRNAs which are found to be drought responsive. And even they have made attempt to identify the molecular targets of those 3 miRNAs, but it needs still a lot more efforts. So for to end up with some useful finding. So I think it's worth continuing that experiment further. It's my suggestion to the advisory committee. And the second one, it's really an interesting story, like photoperiod related studies in PGNP. So they picked their choice of genotypes was excellent. They were able to identify some good set of contrasting parents, and then they went for sequencing and identity. They are all common bioinformatics pipeline. So the interesting feature is that they were able to contrast between the 2 insensitive and sensitive ones. They were able to identify 2 genes, so involved in associated with photoperiod insensitivity. It's worth continuing. So if they are able to demonstrate the function of those 2 genes, like CCFT6 and then CCT23, so and then other CCT4 and CCT23. So they were able to shortlist those 4 genes. So whereas one genotype having upregulation of 4 genes, another one was showing upregulation of 2 genes. So if they are able to narrow down their finding, or if they are able to demonstrate the association of the 2 genes with the function, it would be fantastic. Then as a PGNP genome is sequenced, they can go for allele mining and they can directly put them into breeding applications. So both the PhD works, they were well executed. So in MSc, so there were around 7 theses, I think, and the one is really interesting. So study on the allantoin mediated salinity tolerance. They were able to generate substantial preliminary information. They were able to show that application of this allantoin, so it was found to alleviate salinity induced damages. So it's having potential for translation into a technology if they are able to fine tune the application of allantoin, and then if they are able to dissect out the pathway involved in the accumulation of allantoin in rice, then it will lead to manage the salinity stress. So in a much better way than what we, because no other major QTLs are available for manipulation of salinity tolerance in rice. Other 3-4 studies were characterization of genes like MBF1, calmodulin, and then the hydrotropism related gene, and the cytokinin oxidase or cytokinin dehydrogenase. So the students have carried out the experiment systematically. They were able to clone the gene, but still long way to go to validate them functionally. So another interesting area is that exploiting the BT diversity for VIP3 genes. So the team has carried out the work very systematically. They have done PCR analysis, but the missing link is that bioassay. So if they are able to, they were able to identify 2-3 novel variants of VIP3 gene family, VIP3AA and the AF, and even they were able to identify strains having mutations in the domain 3. So it's a very useful finding or interesting observation. So if they are able to relate it with bioassay or toxicity, so then it will have a lot of commercialization potential. So overall, as presented by Dr. Patnaik, so they were able to show very impressive track record of 86 papers, and even the placement of the students was very impressive. So I congratulate and I thank everyone in the division for taking such hard effort so to convert all the research findings into papers. And I request the team or the guides so to take these leads forward to convert them into technology. And then in physiology, so there are only 6 students, 5 MScs and 1 PhD. And the PhD student, so he worked on a very hot topic like nitrogen use efficiency, one of the burning topics or need of the hour in almost all the cereals. So they have conducted very systematic analysis of RT genotypes, and they were able to show some very, very interesting correlation between the pedigree of those lines and their nitrogen responsiveness. So those cluster of genotypes in one cluster originated from drought-tolerant donors. They were able to exhibit so the better nitrogen use efficiency than the genotypes originated from the other lineage, that is drought-sensitive ones. It's a very interesting observation. It's worth following. And the missing links are light. So they did not present any co-localization analysis between the transcriptome studies and their QTLs, nitrogen use efficiency related QTL. If they would have performed this analysis, so they would have been able to narrow down the candidate genes for further selection or breeding applications. So, but they have 2-3 major leads, so which needs further attention or continuation. So in MSc, all the 5 MScs say they have 2-3 very interesting preliminary leads. So one is on manipulation of stay green trait, another one is this on trait. So in alleviating heat-induced yield losses. So it's worth following these 2 leads generated by the master's research. And under elevated CO2, so the team has pointed out 2 observations, very, very interesting. One is that elevated CO2 is delaying the germination in the subsequent generation. So they have also observed it very critically. So if they are able to validate this observation over years or over season, so then this will give a preliminary leads or ideas for the scientific community to work on this area. And the second lead they have obtained is that, so the reproductive stage N application, it helps in increasing the grain protein content in wheat. So this is also having a lot of potential. So if you are able to validate it across environment or by further experiment, so it would be useful to the both industries and as well as for the consumers. In the biochemistry division, I could see the dedication of both the guides and the students. So almost all the studies are very impressive. So especially the PhD student, he tried to optimize the non-destructive method of phenotyping for the metabolite composition. It's an interesting area. It's an emerging area. in this world of artificial intelligence and machine learning. So the preliminary leads developed by the student so would serve as a base for developing the predictive models for quantifying the secondary metabolites in that are pearl millet especially. So they were able to see or record more than 80% correlation for most of the metabolites they have observed, and the student has come out with two very impressive publications. So one in food science and technology, and the second one is in food composition and analysis. Both of them are in the Science Direct publication. Here also I could see, so they have carried out nice analysis. They have surveyed number of compounds, but they have missed 2-3 major very important compounds which are very tricky to analyze or which is very difficult to analyze. One is like goitrogenic glycosylflavones, and then carotenoids and tannins. They have done analyzed all the carbohydrate fraction, proteins, lipids, etc. So if they could have carried out the analysis of these compounds, which are very difficult to analyze biochemically, so then it would be useful for the scientific community to use this near infrared spectroscopy for quantifying these compounds. I congratulate the team. And in masters, so there are 5 theses. So 2 on controlling quality, bioavailability, and the functionality of nutrients in different food matrices, and 2 theses on enhancing the nutritional and the keeping quality through processing. All the 5 are well executed. So in the first one, so the student has tried to deciphering the influence of food matrix state of composite flour and nutrient quality and bioavailability. So they tried or they compared 6 different nutrimixers. So initially it looks like a common regular research, but so the final observation made by the student was very impressive. Like they were able to identify one composition or one mix, so having potential to replace the chapati making duff, wheat duff, having potential. They did all the analysis and they found that so the Nutrimix 3, I think, yes, having great potential to act as a sustainable alternative to wheat. So, but here also the missing link is that I request the guy to compare their technology or the product with the commercially available mixer. So that will help them to get any license or patent in future. And then they can very well take it for household trial, especially in rural areas. And the second study was very interesting clinically. They tried to study the interaction between starch and lipid, so in reducing the inherent glycemic potential. Very, very interesting study. Though they compared the interaction of rice and pearl millet flour with 5 different oils, and they found that especially the mustard oil in combination with both pearl millet and rice having ability to lower the glycemic potential. So a lot of clinical significance is there. I think it can be protected right now. They can plan for protecting their concept. So, but it needs a lot of refinement, and they can go for testing the glycemic index of the food combination through clinical trials. And then other 2 studies, like other 2 potential research, so that has led to development of leads for translation. One is understanding the effect of probiotic fermentation. I can say this is one of the emerging areas. Like people are looking for alternate to cow milk, and soy milk is one of the potential alternate. But there the major problems are like so flavor, and then the taste, odd taste with that soy milk. And here the student has demonstrated that some of their combinations are the treatments, like treatment with the probiotic Vaisala and then pre-germination. So it reduced the level of anti-nutritional factors. So it's an interesting observation, and they were able to see so the reduction in the off labor also. So, but the taste was not up to the mark. So here at that, I suggest that so they can, they need not have to aim for 100% soy milk. So if they blend it with normal dairy milk or the cow milk, so at you can reduce the requirement of cow milk. You can come out with a new product which will help you to improve the acceptability among the consumers. So, and the another one is like utilization of rice bran, which will add value to the rice crop as well as increase the farm income. They were able to see like infrared treatment was found to be effective in stabilizing the rice bran oil quality. So very useful finding to the industry. So they can compare it against the available technologies, and these leads can be taken forward. So for further research as well as for technology transformation. So I think I have covered all the aspects. So all the 21 studies or thesis research so were well executed. So once again, I congratulate the students as well as the advisory committee members and the chairman for their dedication and their maximum care in executing the work. Thank you very much, sir. Thank you once again for the opportunity given. Thank you.
Thank you, Professor Ravindran, for that excellent summary and the comments and suggestions for taking the leads forward. And that's a lesson for a student and also for the chairperson. And we will try to incorporate it in our program. Thank you so much for being here and sparing time. And that shows your involvement, how critically you have gone through all the presentations and come out with very straightforward analysis and recommendation of each topic.
Thank you, sir.
Thank you so much.
Thank you, sir.
Thank you, sir.
Sir, on behalf of Institute and my own behalf, I am grateful to Dr. Amravindram, Professor and Head, Plant Biotechnology, TNAU, for gracing the occasion and sparing valuable time. Sir, I was going through your biodata, sir, from internet, and you obtained as many as 17 externally funded projects from different national and international organizations. This is something, sir, admirable, sir. Sir, the kind of pioneering contribution you made in the area of molecular biology and biotechnology, I am sure your presence as well as your elaborate analysis, positive review, and critical comments, this inspired our faculty as well as students, and that will be very helpful in shaping our future research. Thank you. Thank you very much, sir.
Thank you, sir. Thank you, sir. Once again, I thank the Director IRI for providing me this opportunity. And my mentor also is my right from my joining at TNAU during 2006-07. I'm working with Dr. A.K. Singh, Dr. N.K. Singh, and Dr. Viswanathan, who are all my inspirations. Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir. Thank you.
Let us proceed to the next
We will proceed to the next session.
If Professor Hariram has joined.
Yes, sir. Welcome, sir. Sir, I have seen him, sir. He has joined, sir. Yes, sir, he is there, sir. Good evening, sir.
Sir, you are muted.
Muted, sir. You are muted, sir.
Yeah, it's okay. Now.
Yes. Good evening, Dr. Sahab, and hearty welcome. Thank you for kindly accepting our invitation for chairing this session. Thank you so much.
My pleasure, sir.
Please proceed, Dr. Rasmi.
Yes, sir. Good evening to all of you. Most esteemed chairperson of this session, which is session 5, Dr. Harihar Ram, our Honorable Director, Dr. A.K. Singh, all the project director, Dr. Man Singh, heads of the division, professors, faculty, students, all those who have really joined this 5th session of 60th Convocation Week program. All of you are most welcome to join this session. So you will be all be very glad to know that for this session, we have Dr. Harihar Ram, former professor and head Department of Vegetable Science, G.B. Pant University of Agriculture and Technology, Pantnagar, well-known researcher in the area of vegetable sciences. So we are very glad on behalf of Institute, on behalf of Director IRI, all of us warmly welcome you to this session, which is on the presentation on the significant educational achievements under the School of Horticultural Sciences. So the School of Horticultural Sciences has 4 different disciplines. They are floriculture and landscaping architecture, then fruit sciences, post-harvest technology, and vegetable science. So total there are 43 degrees to be presented. 43 students' significant achievements will be presented by the professors of these disciplines. And out of these 43, there are 27 MSc students and 16 are the PhD students. So based on the significant achievements of these students, professors will make their presentations. And let me share with you, this will not be the total research activity scenario of the School of Horticulture Sciences. So particularly the presentation will be based on only the thesis of the students who submitted the preceding year, and they will be awarding degrees. They will be awarded degrees during this 60th convocation to be held on 11th February 2022. So with these few initial remarks, I welcome once again all of you, and I now would like to ask our Honorable Director to kindly introduce Dr. Harihar Ram to all the joinees.
Thank you, Dr. Rasmi. My hearty welcome to Professor Harihar Ram, one of the doyen of vegetable science and soybean research in the country. His contributions are enormous, cannot be described in words. And he has been serving earlier in different capacity as a resource person on one or the other program of IRI. I would like to extend a warm welcome to him. If you look at his academic credential, this is something that every student would aspire, and particularly those who are familiar with UP board would know that how difficult it was, to top the UP board with number of students running to something like 12 lakhs to 15 lakhs every year in the examination. And this was the time, like this must have been long back, somewhere in the could be 60s or so. Dr. Harihar Ram, with an excellent academic career, he got first position in UP board in intermediate examination, first position in Gorakhpur University in BSc Agriculture, first position in Masters and PhD program at Pantnagar, first position in ICR Senior Fellowship, and completed his Masters and PhD plant breeding from Pantnagar and postdoctoral program from Germany. So what a terrific credential that Dr. Harihar Ram has as a student. And then he served for 33 years at Pantnagar University in various positions, including the head vegetable science, joint director vegetable research. But his contribution to soybean improvement and vegetable improvement has been remarkable and phenomenal. So after serving and superannuating from Pantnagar, he served as vice president and chief of R&D in private sector. And there also he made enormous contribution. He served as a consultant to FAO also in Myanmar, Libya, and DPR Korea. Released 9 varieties of soybean, 26 varieties hybrids of vegetable crops. And when he joined private sector, you can see that might have been 6 to 7, 8 years period. He, helped the commercialization of 39 cultivars and hybrids of vegetable crops in seed industry. So he's also written 18 books and 260 research papers, and guided 25 masters and 26 PhD students. His 2 books, particularly Vegetable Breeding and Crop Breeding and Biotechnology, are immensely popular among the students in all the state agriculture universities. It's our pleasure to have Dr. Harihar Ram to chair this session on the School of Horticultural Sciences, where we are going to have the presentation by the professor of the significant research achievements by the students of the respective divisions. Yesterday we had presentation of the best student from each division. They were competing for the various medals, and equally important research contribution by the students who could not compete there. The work is will be presented by the respective professors of the discipline. One announcement for the students who are with us, and I'm sure there are many. We have total 539. So you have the announcement for the best student medal, Professor Real Chopra NABARD medal, and IRI medal that will come up after all the presentations are over. We are just doing some exercise. It is almost done, and it will be announced after the presentation of the School of Social Science. So kindly be with us, bear with us, and also learn what your colleague friends have done. Thank you very much.
Yeah, thank you, sir. Thank you very much. So we are going to start the presentation of School of Horticultural Sciences. And this is also very important school, one of the very important school of the institute. Horticulture accounts for 30% of India's agricultural GDP, from maybe 15 to 20% of crop area. It has a lot of meaning because it is very important for rural economy. So with this word, as Dean Madam made mention that there are 4 disciplines, and with the kind permission of Chairman Sir, can we take a first presentation? Sir?
Yes. Am I audible, please?
Yes, sir. Yes, sir.
Okay. So please, before that, I express my sincere thanks to Professor A.K. Singh for very kind words, Madam Rasmi Agrawal, and all of you for giving me an opportunity to be here. And for me, it is all the way learning and learning and learning and refreshing my memory. So thank you very much to all of you. Please go ahead.
Thank you, sir. Thank you very much. So first presentation will be from Floriculture and Landscape Architecture. And Professor Kanwar Pal Singh will present.
Respected Chairman of the session, Honorable Director IRI, Dean and Joint Director Education IRI, Head of Divisions, Professors, and dear students. I am going to present significant education achievements for the year 2021 for the discipline of Floriculture and Landscape Architecture. This is our position of faculty and students at IRI New Delhi and IHR Bangalore. And there are 5 students of MSc and 2 PhD who are going to receive their degree during this convocation. Coming to the research achievements in theme number 1 on improvement of flower crops, the work on marigold was carried out by Ms. Adiga Amla, taking 2 species of marigold, that is erecta and Tetraspatula, spatula, with the following objectives, and she took 20 varieties of marigold belonging to both the species, and the experiment was conducted under polyhouse and outside polyhouse conditions to compare these varieties in both the growing environments. And observations were recorded on morphological parameters, physiological parameters, biochemical parameters, and image-based traits. Study revealed that electrolyte leakage rate was found minimum in genotype FW4 of African marigold and this photosynthetic rate was found maximum in FW4 and Isar Jaffrey, followed by Isar Beauty. And proline content was also found maximum in FW4 and Isar Beauty genotypes. And SOD activity was also found maximum in AFW4 and Isar Beauty genotypes in both the experiments. And these are pictures of some image-based traits which were taken for high-throughput phenotyping of cold-tolerant genotypes in marigold. With regard to salient findings, based on various morphological, physiological, and biochemical parameters, the most cold-tolerant genotype was found AFW4 in African marigold and Isar Beauty in French marigold. Image-based traits also confirmed more cold tolerance in genotypes AFW4 and Isar Beauty. Another work was conducted on development of in vitro regeneration protocol in apetalous male-style lines of marigold by Mr. Nandish Ganiga with following objectives. And he took 3 milk style lines and 2 type of explants and various growth regulators in different combinations. And this is the salient findings. A study revealed that MS medium supplemented with 1.5 milligram per liter of TDZ and 0.2 milligram per liter of NA gave better results with regard to culture establishment and early shoot initiation. Regarding shoot proliferation, MS medium supplemented with 0.5 milligram per liter N18 and 0.1 milligram per liter Na was found optimum. This study was conducted on chrysanthemum by Mr. Sunil Kumar at IHR Bangalore with the following objectives. And these are the genotypes he took for his study. And this is the technical program of various experiments. And this is the field view of his experiment. And regarding salient findings, the genotype red stone showed maximum percent of viable polar grains. And among the genotypes studied, 11 genotypes were found to be resistant to white rust. Out of total 54 half-ship progenies screened, 18 half-ship progenies showed nil disease index. Coming to the next theme on crop protection, work on characterization and management of phytoplasma in rose phenotypes was carried out by Rene with the following objectives. And for this work, survey was conducted in 4 states of India for phytoplasma detection and collection of samples of roses, weeds, and insects. And these samples were subjected to an extraction and PCR analysis. Based on that, cultivar MS Randhawa was selected after confirmation of the presence of phytoplasma for its management using antibiotics. And for that, 3 antibiotics with various concentrations were used under in vitro and in vivo conditions for phytoplasma management. Regarding salient findings under Objective 1, phytoplasma suspected symptoms of phyllodes, flower malformation, little leaf, and flat stem were recorded in 16 rose genotypes. Phytoplasma association was confirmed in all the 16 rose genotypes using different primer pairs. Regarding the salient findings in Objective 2, leaf hoppers collected from New Delhi and Uttar Pradesh were tested positive with the 16 SR2D subgroup of phytoplasma. And with regarding this weeds, 5 species of weeds collected from New Delhi were identified as hosts of 16 SR1 and 16 SR2 groups. And 4 species of weeds collected from Uttar Pradesh identified as hosts of 16 SR2 group. Regarding salient findings under Objective 3, all the concentrations of oxytetracycline were found effective. However, concentration of 60 milligram per liter was observed to be the best in terms of plant growth regulators and development. This study on lilium was carried out by Ms. Saraswati with the following objectives. And this is the technical program of her study. And this is the crop grown by her under SEDNet for the research work. And regarding salient findings, among various concentrations of organic manure, FIM at the rate of 25 ton per hectare performed better for vegetative growth parameters, quality flowers, bulbs, and bulblet production. Among various concentrations of inorganic fertilizers, plant sprayed with NPK at the rate of 160, 120, and 110 kg of per hectare performed better for most of the parameters. Coming to the next theme on post-harvest management, this work on marigold was carried out by Mr. Grace with the following objectives. And this is the technical program of his study. And these are the view of the different experiments where the loose flowers of marigold are kept after giving treatments. Regarding salient findings, among the preservative used, boric acid treatment at the rate of 4% was found to be the best as compared to control, which gave maximum shelf life under ambient and cold storage conditions. Among packaging materials used, shrink wrap packaging material was found best for enhancing the shelf life of loose flowers of marigold. Next study on this post-harvest management was carried out by Ms. Priya Bibi in tube rose with the following objectives. And this is the technical program. And these are the loose flowers of tube rose after giving pre-treatments. With regard to salient findings, the flowers treated with boric acid at the rate of 4% and packed in muslin cloth bags exhibited maximum moisture content, floret diameter, shelf life, and SOD activity. And with regard to packaging material, among packaging materials tried, HDPE was found to be the best, which resulted in maximum moisture content, floret diameter, shelf life, and SOD activity. These are some training programs which were organized by our division, and this is the placement of our students and number of research papers published by the division during the year. This is the list of awards and honors received by our faculty members, and this is the list of awards and honors received by our students. And our students also participated in sports and other extracurricular activities of the division. These are our PG labs where our students are doing research. These labs were recently renovated. I acknowledge the support and guidance received from Director IRA New Delhi, Dean and Joint Director Education IRA New Delhi, Joint Director Research IRA New Delhi, Joint Director Extension IRA New Delhi, heads and professors of IRA, faculty members, and students. Thank you. Thank you one and all.
Yeah, thank you very much, Professor Kanwar Pal Singh, for your very good presentation. Sir, Chairman, sir, if you kindly permit, then we will go for next presentation. And at the end of all presentation, we will take your comments.
That is fine, please.
Okay, sir. Thank you, sir. So next presentation will be from Fruit Science by Professor Manish Srivastav.
Respected dais, other dignitaries present in this session, IRI faculty, students, ladies and gentlemen. I'm going to present the significant educational achievements pertaining to the discipline of fruit science for the year 2021. These are the mandates of the division, and currently we have 21 faculty at IRI, including IRI Jharkhand. At IHR Bengaluru, there are 10 faculty. Total 49 students are on roll at IRI, including IRI Jharkhand, out of which 17 are for MSc degree and 32 are PhD students. At IHR, there are 14 PhD students on roll. This is the list of 9 students, those who are going to have their degree in this convocation. Out of 9 students, 3 are for PhD degree and 6 are for MSc degree. The research work carried out by PG students are grouped under different themes. The first theme is genetical studies and molecular characterization. Mr. Sridhar Ramachandra, a PhD student, worked under my guidance, and his PhD thesis topic was QTL mapping for fruit quality traits in mango. And the objectives were to study the phenotypic variability for fruit quality traits in the mapping population of mango and to identify the QTLs governing fruit morphological and biochemical traits in mango. He used 85 biparental progenies obtained by crossing 2 contrasting parents, Amrapali and Sensation, and these progenies as well as the parents were phenotyped for 27 fruit quality parameters for 2 years. For genotyping, he utilized SSR and SNP markers, and for construction of genetic linkage map, he used joint map version 4.1, and for QTL analysis, he used map QTL version 5.0. Mr. Sridhar observed significant variations for the traits like fruit shape, fruit weight, peel thickness, TSS, beta carotene, firmness, peel and pulp color in the mapping population. For genotyping of population, he used SSR markers and he screened 558 SSRs for parental polymorphism, out of which only 28.67% were polymorphic, but he could not generate the linkage map using the SSR marker due to insufficient polymorphic markers in each segregation group. As an alternative, he used SNP markers, and finally he could map 492 SNPs across the 20 linkage group as per the haploid chromosome number of mango. The cumulative genetic map distance was 4974.1 centimorgan. Utilizing the genotypic and phenotypic data, he attempted QTL analysis, and initially he identified 10 QTLs, out of which 5 QTLs were stable over the years, and they were located on chromosome number 11, 15, and 16. The traits were fruit length, fruit width, stone width, fruit peel brightness, fruit pulp color brightness. And the information generated on QTLs need to be validated in diverse mango population, and that can be further used for genomic assisted breeding in mango. Another student, Mr. Kiran Kumar GN, worked under the guidance of Dr. M.R. Dinesh, and his PhD thesis topic was evaluation of putative plidy progenies of guava. The objectives were to study the variability in plidy manipulated progenies, to identify progenies having dwarf stature, and to identify progenies having fruits with desirable traits. These are the details of the materials and methods adopted by him. He could get different numbers of colchipliers under different treatments, and finally he got 551 progenies or colchipliers which were evaluated further. He observed that the seed-treated progenies of Arca-Kiran showed maximum variation for the vegetative characters. However, the seed-treated progenies of Arca-Mridula showed maximum variation for fruit characters. Finally, he concluded that plidy manipulation can be considered as a very good approach to induce large-scale variability in guava. And he also shortlisted some progenies having different plidy levels, and they can be used as a pre-breeding lines. And he also identified progenies having desirable traits. One of our MSc student, Mr. Kiran, worked under the guidance of Dr. Avtar Singh. His MSc thesis topic was morphogenetic characterization of second generation colchipliers of sweet orange cultivar Mosambi. The objectives were to characterize second generation colchipliers of Mosambi with respect to morpho-physio-biochemical traits, and also to use cytological and molecular tools for characterization of the second generation colchipliers. These are the details of the materials and methods adopted by him. He observed that the second generation colchipliers developed from the putative tetraploid branches of first generation showed both stimulatory and inhibitory responses for the various traits, and there was significant alterations in terms of leaf characteristics, leaf sclerophyly, stomatal aberrations, photosynthetic pigments have been observed among the colchipliers. Based on the molecular and cytological analysis, he could identify 8 stable tetraploids, and these selected solid tetraploids of Mosambi may serve as useful resource for the future breeding program targeted to develop triploid Mosambi. Mr. Sandeep, MSc student, also worked in the same area under the guidance of Dr. Avtar Singh, and his MSc thesis topic was morphogenetic characterization of second generation Kinno Mandarin colchipliers. The objectives were to characterize second generation colchipliers of Kinno Mandarin with respect to morphological, physiological, and biochemical traits, and cytological and molecular characterization of the second generation Kinno Mandarin colchipliers. These are the details of material and methods adopted by him. He also observed that the second generation colchipliers indicated both stimulatory as well as the inhibitory responses for the various traits, and based on overall characterization of colchipliers, he identified 11 putative tetraploids, out of which 6 were solid. Mr. Shivam worked under the guidance of Dr. Kanhaiya Singh, and his MSc thesis topic was morphological, physiological, biochemical, and molecular characterization of progenies of Olure mango. And the objectives were to confirm the zygotic saplings through molecular characterization and to evaluate the progenies of Olure on the basis of morphological, physiological, and biochemical parameters. These are the details of material methods adopted by Mr. Shivam. He used 4 EST SSR markers for screening of open pollinated progenies of Olure. Based on research findings, he concluded that LMMA2 and LMMA8 SSR markers were informative for ascertaining the zygotic and nucellar origin of open pollinated Olure progenies, and out of 90 progenies screened, he could ascertain the zygotic origin of only 28. And based on the morphological, physiological, and biochemical parameters, he identified 6 progenies as dwarf one and 5 progenies as vigorous one as compared to the maternal Olure. One of our MSc student, Ms. Rutuparna Senapati, worked under the guidance of Dr. Madhubala Thakre, and her MSc thesis topic was understanding the basis of pulp color in black guava. She also presented for IRI Merit Medal day before her study, and the objectives of her study were estimation of metabolites responsible for pulp color in black guava and to characterize the genes involved in metabolite synthesis in black guava. These are the details of material methods she used during her study. Based on the LCMS analysis of black guava, she observed that there are 3 different anthocyanins which are responsible for the purple pulp and peel color of the black guava, and they were delphinidine 3-glucoside, petunidine 3-glucoside, and cyanidine 3-glucosides. From the research findings of her study, she concluded that in guava, the pulp color is due to combination of more than one pigments that may be carotenoids, anthocyanin, and lycopene. And the final color depends on the relative proportion of these pigments. And this stage in guava for pulp color, the statement pigments in proportion is correct. Second theme is crop improvement and character association. Mrs. UVCG Mary Grace, a PhD student, worked under the guidance of Dr. M.K. Verma, and her PhD thesis topic was identification of grape genotypes tolerant to berry cracking. And the objectives were characterization of grape genotypes for morphophysical and biochemical traits, and screening of grape germplasm for cracking incidence, and association of characters with reference to the berry cracking. Used 29 grape germplasm from different origin having different parentage, and these are the methodologies she used during her research work. She observed that the genotypes having large compact bunch and round bold berry had more berry cracking incidence, and the physiological activity were more in cracking sensitive genotype. She also identified some of the tolerant and susceptible grape genotypes based on berry cracking ratio. Finally, she concluded that the berry cracking associated traits are bunch texture, TSS, water uptake, and vine physiological activities. One of our MSc student, Mr. Mukesh Shivaran, worked under the guidance of Dr. Nimisha Sharma, and his MSc thesis topic was molecular studies on shelf life in mango, and the objectives were molecular characterization of the selected mango genotypes with varying shelf life using the newly designed primers, and to study the effect of rootstocks on shelf life of mango genotypes. He used 20 mango genotypes having long, medium, and short shelf life, and shelf life specific primers were designed from the ripening nucleotide sequences of the mango. In the second experiment, he used Olure, Kurakan, K5, K3 as rootstock, and sign varieties were Amrapali, Dasheri, Mallika, Pusa Renuma, and Pusa Surya. He observed that the molecular analysis of mango genotypes using shelf life specific primers grouped them according to their shelf life pattern, and based on the molecular analysis it was found that sign varieties grafted on Olure produce fruits having more shelf life. So he concluded that the molecular characterization of important mango genotypes for shelf life trait via ripening related gene specific primers could be utilized for marker assisted breeding in mango. The third theme is crop regulation. Mr. Bhupen Sagore worked under the guidance of Dr. Kanhaiya Singh, and his thesis topic was effect of plant growth regulators on hastening embryo maturation and fruit quality of papaya variety Pusananna. The objectives were to study the effect of etheryl, abscisic acid, and methyl jasmonate on in vivo embryo maturation and in vitro regeneration, and to find out the response of these PGRs on fruit quality parameters at ripening stage. These are the details of materials and methods adopted by him during her MSc research work. He took Pusa Nanna variety, and the PGRs were etheryl, abscisic acid, and methyl jasmonate. Mr. Sagore observed that the etheryl 120 ppm at 90 days of flowering was most effective for in vivo embryo maturation. However, the treatment of methyl jasmonate was effective for emergence of maximum number of roots and longer root length. He concluded that the etheryl 120 ppm was effective in hastening embryo maturity and improving the fruit quality of Pusa Nanna papaya, followed by methyl jasmonate. This is the list of the awards and honors conferred on our faculty. Dr. Anil Kumar Dubey was honored for the Fellowship of National Academy of Agriculture Sciences. Dr. Radha Mohan Sharma acted as editor in the Journal of Horticulture, and Dr. Madhubala Thakre and Dr. Chaulesh Kumar got Best Poster Awards. Two of our students, Mr. Mukesh Shivaran and Bhupen Sagore, secured first and second rank in All India Entrance Examination conducted by ICR. And as far as the placement of our student is concerned, Ms. Preeti Singh was appointed as assistant professor at BAU, Bihar. Mr. Sunil Kumar joined ARS services, and he is posted at ICR National Research Center on Lychee. And Mr. Hanuman joined as assistant horticulture officer, Government of Karnataka. Thank you very much for your patience hearing. Thank you one and all.
Thank you very much, Professor Manish, for very nice presentation. And we will move to the next one, to poster-based technology. Professor Ramastra will make presentation.
Respected Chairperson, dignitaries, Head of Divisions, Professors, Faculty and my dear student, I am here to present the significant educational achievements of post-harvest management discipline for the year 2021. Mandate of Food Science and Post-Harvest Technology Division is to undertake research on the integration of production with post-harvest management practices, development of appropriate processing system for value-added products, human resource development, transfer of technology along with consultancy. Presently, we have faculty strength of 7 and 34 students are on roll in the division. During this convocation, 12 students will be receiving their MSc and PhD degree. The MSc and PhD students take their research in the area of post-harvest management of fresh fruit and vegetable, processing of horticultural and arable crops. So moving to research area of post-harvest management of fresh fruit and vegetable, Ms. Archana Janamati has conducted her PhD work on management of chilling injury, shelf-life and post-harvest diseases in mango with 3 major objectives. In her methodology of experiment consists mature mango fruit treatment with different concentration of Guava, glycine vetenin, Pseudomonas nitida and carbendazim. Out of this experiment, she concluded that Guava application at the rate of 10 millimolar has effectively controlled chilling injury during storage at 7 degrees Celsius storage temperature. Glycine betaine treatment at the rate of 15 millimolar retained desirable quality of fruits even at ambient storage temperature of 32 degrees Celsius for 12 days. Another PhD student, Ms. Uma Prajapati, taken off her research on studies on the influence of UVC and chemical elicitors on quality and shelf life of bitter gourds with 3 major objectives. For this, she treated the immature bitter gourd fruits with different doses of UVC, salicylic acid, putrescine and calcium lactate and stored the fruits for 20 days at 7 degrees Celsius storage temperature. In results, she concluded that UVC 40 minutes treatment was best for retention of nutraceutical compound and enhancement of stress-related metabolites. Salicylic acid 10 millimolar and putrescine 3 millimolar proved best in extending the shelf life of bitter gourds up to 20 days under cold storage. Calcium lactate 100 millimolar was rated best in retaining the firmness, reducing the PME activity along with retention of post-harvest quality attributes during storage. Gene expression study shows highest DDCT value of MCCS1 and MCSE gene in putrescine treated bitter gourd fruits on 20th day of storage. Mr. Karan Singh Dhami taken up a study on post-harvest quality regulation of quinomycin through exogenous application of pre and post-harvest application of methyl jasmonate with 3 different concentrations, use of lemongrass oil and potassium permanganate in packaging system. In outcome, he found that foliar application of methyl jasmonate 20 days before harvesting showed better response over post-harvest application. In both the pre and post-harvest application, methyl jasmonate at the rate of 0.5 millimolar concentration was found most effective in maintaining the quality of quinomycin under both the temperature, that is 2 and 6 degree centigrade storage temperature. Active packaging system with 5 milliliter lemongrass oil in 20 kg CFB box extended the shelf life of quinomycin fruits for 40 days at 6 degrees Celsius storage temperature. Mr. A.J. Sachin taken up study on extension of storage life of guava using ethylene inhibitors and its integration with modified atmosphere packaging with 3 objectives. For this, he treated the mature guava fruits with different ethylene inhibitors including 1-methyl cyclopropane.
In results, he concluded that among ethylene inhibitors, 1-methyl cyclopropane at the rate of 500 ppb was found effective in extending the storage life of Arca amradula Guava fruits. Cryobake packaging material PD961 with and without microperforation effectively maintained optimum in packaging modified condition at 12 degrees Celsius temperature. The expression of expansin and SCC synthase gene were rapidly inhibited by ethylene inhibitors, and thus they may be a good candidate for manipulating softening in guava through recombination DNA technology. Mr. Devesh Kumar conducted his study on effect of pre-storage polyamine treatment on quality and shelf life of pointed guard with two objectives. In methodology, he first optimized the doses of putrescine and spermidine in first trial and later on treated the pointed guard fruits with optimized concentration of putrescine and spermidine and stored the fruits at ambient and cold storage for 12 and 21 days respectively. He concluded that putrescine at the rate of 1 millimolar and spermidine at the rate of 3 millimolar retained most of the desirable quality characteristics of stored pointed guard. Putrescine 1 millimolar and spermidine 3 millimolar successfully extended 6 days extra shelf life over control at cold storage condition. Mr. Omar Alazil, a foreign student, conducted studies on nutritional profiling of apricot and development of osmodehydrated products with 3 objectives. For this, he chosen 6 apricot varieties for osmotic drying with different energy levels of microwave and ultrasonicator and recorded different quality parameters of dried apricot. In present study, he found that apricot cultivars CITH-A2 and CITH-A1 were found most suitable for the preparation of osmodehydrated apricot. Kernel of CITH-A2 followed by CITH-A3 exhibit most favorable characteristic for preparation of other value-added product. ALP 260 gas packaging material was found more suitable for storage of osmodehydrated apricot at ambient condition up to 6 months. Mr. Hanvesh Gowda taken up a study on valorization of pumpkin to enriched crisps with 2 objectives. Mr. Gowda used mature fruits of commercially important pumpkin genotype and sliced them and impregnated with calcium lactate and calcium chloride by vacuum impregnation system. Impregnated slices were subjected to microwave drying, packaging and storage study. In result, it was concluded that calcium lactate was best for slice impregnation over calcium chloride. Genotype DPUK06 was found superior over others cultivars. In another study, Mr. Asuja Vian studies on overripe bitter gourd oleogel for nutritional enhancement of cookies with 3 major objectives. In methodology, he taken up ripe bitter gourd fruits, extracted pulp mixed with different proportion of canola oil and ethyl cellulose and finally converted into cookies. In result, he concluded that ethyl cellulose could be mixed with canola oil at concentration above 4% to form oleogel with solid-like properties which could be utilized as a hydrogenated fat substitute in cookies. Without affecting the sensory qualities of cookies, up to 12% bitter gourd powder can be integrated in the oleogel and up to 40% hydrogenated fat can be replaced in the cookies. Tippeswamy B conducted a study on extraction and enhancement of functional stability of wheat root betalains with 2 focused objectives. In methodology, he compared 2 varieties for their betaline extraction suitability, finalized protocol for betaline extraction and evaluated stability of betaline with and without black carrot co-pigmentation. In this study, he found that beetroot crimson glove variety was found superior for color extraction over the Detroit dark red. 70 degrees Celsius extraction temperature along with 4 minutes extraction time and 1:3 solid to solvent ratio was found to be best for higher battalion recovery. Co-pigmented battalions with 80% black carrot pigment showed higher stability in comparison with native pigment when exposed to temperature more than 60 degrees Celsius and light intensity of 2400 lux. In another study, Mr. Sajil Ahmad conducted experiment on assessment of onion genotype for dehydration suitability and functional quality with 3 focused objectives. In methodology, he has taken 8 genotypes for assessing their processing suitability. The best rated genotype Pusa sova was subjected to different unit operations of dehydration and storage study. In findings, he concluded that Pusa sova was found most suitable for dehydration purpose. Sodium chloride 5% pre-treatment and 60 degrees Celsius drying temperature retains most of the desirable quality parameters of dried onion slice. 200 gauge SDP proved superior over 200 gauge DPE packaging film. Sanjit Jadhav conducted trial on evaluation of microwave recirculatory air drying of large cardamom with 2 objectives. For this, he chosen Ramsay and Varlange varieties of cardamom and dried them with microwave and recirculatory air dryer and recorded quality parameters like color kinetics, texture analysis, qualitative and quantitative analysis of volatile oil. Out of this experiment, he summarized that drying times of microwave drying 30 to 300 minutes were significantly lower compared to recirculatory air drying, that is 280 and 840 minutes for both the varieties. The maximum oil yield was obtained at 100 watt power level for both Ramsay and Varlange cultivars. Mr. Om Prakash studied on twin extrusion of formulate with 2 objectives. In this experiment, he has taken 4 cultivars for selecting the best one for milling and noodle preparation. The best found variety Pusa Composite 1201 was subjected to different extrusion unit operation and quality parameters of finished noodles were recorded during storage period. Out of this study, he concluded that Pusa Composite 1201 variety was found best for noodle preparation. Guargum 2% was proved the best additive for subsequent improvement in cooking quality of pearl millet noodles. The substitution of pearl millet flour with 50% defatted soya flour yielded noodle strength of desirable characteristic and increased hardness value. The noodle strength 50:50 blends of pearl millet flour and defatted soya flour were found consumer safe up to 3 months of storage period. Coming to the awards and honors, our faculty and students have conferred with several prestigious awards and honors during year 2020 and 2021. Also, the placement record of our student has remained impressive during the year under report. Finally, I would like to thank each and everyone for patience hearing.
Yeah, thank you very much, Professor Ramastra, for your nice presentation. Now we will go for the last discipline of this school, and Professor R.K. Yadav will make presentation on behalf of vegetable science. Thank you.
Namaskar to everyone, respected resource person, distinguished scientist and student. I am going to present before you the significant academic achievement of postgraduate student of Division of Vegetable Science. The major mandate of the division is to do the strategic and applied research for improvement of vegetable crops. We have also mandated to teaching and guidance of postgraduate student and imparting training for human resource development. Dissemination of information on recent advances in vegetable crops is another mandate of this division. The major thrust area of this division is to develop varieties and hybrids for high yield, high nutritional quality, and resistant to different biotic and abiotic states using conventional and molecular approaches. At present, we have the faculty around 36. This includes IRI, IIHR, IRI Jharkhand. This year, the number of PhD students 6 and MSc 10 are going to receive degree. The students worked under different aspects like molecular breeding, mapping and tagging of genes, genetic diversity, heterosis breeding. The first student, Siddharth, who conducted his PhD thesis on the joint multiple family linkage analysis for resistance to watermelon, bird necrosis virus, and fruit quality testing. And for this study, to understand the genetics of fruit quality traits in watermelon, they developed the pre-breeding lines from wild watermelon genotypes citron, and these pre-breeding lines were used to cross with cultivated lines, and their population were developed and genetic was studied, and it was found that most of the fruit quality traits were governed by one or two genes, and suggesting the simple inheritance. For various quantitative traits, QTLs on different chromosomes were identified. In candidate gene analysis, several putative genes governing various traits at different regions were also identified. From metaQTL analysis, 3 metaQTLs were identified on chromosome number 2, chromosome number 3, and chromosome number 7. From these metaQTLs, the genes were putative genes or candidate genes were also identified for resistant to insect and viruses. The breeding for combined resistant to bacterial wilt and root knot nematode in tomato was studied by Mr. Vinod Jatov. To develop multiple disease resistant line having resistant to bacterial wilt and root knot nematode, they first identified a resistant line for both these traits, and they also screened molecular markers tightly linked with these two diseases. Further, the advanced lines were developed by backcrossing, and at every stage of the backcross, the presence of resistant gene was confirmed with tightly linked molecular markers. And finally, they developed 4 lines which were found the presence of both the QTLs for BWR12 and BWR6 along with the presence of MI genes, and these lines showed resistant to both the diseases. And advanced lines developed from this study can be further used for pyramiding of genes for development of multiple disease resistant lines in tomato. Genetic analysis of male steroid mediated 3-way crosses for yield quality and resistant to purple blot disease in onion was carried out by Mr. Ajay Kumar Pandav. They found that the 3-way hybrid Arka Lalima and crossed with Arka Kalyan gave 57.9 ton per hectare yield, and similarly in disease screening, the hybrid Arka Kirtiman and Arka Kalyan was found very good resistant to purple blotch disease of onion. Incorporation of yellow mosaic disease resistant in pole type French bean was studied by Mr. Arindam Das, and he developed a number of crosses and he identified a number. In screening study, they found that the 2 resistant genotypes Arka Arjun and Jade 5058. In inheritance to this resistance, they found that single recessive gene was controlling the resistance to this disease, and the marker PBCOM14 was found linked to horse gram yellow mosaic disease in French bean. Heterosis study on yield and quality traits in temperate carrot using cytoplasmic mail sterility was done by Mr. Hemant Ghemre. He developed 100 hybrids by utilizing 10 CMS lines and 10 inbred. And then performance study, they found that the number of hybrids showing very high yield, and also they have found some hybrids good in nutritional quality, especially KT98 and crossed with PN1 gave 54 ton per hectare yield. Genetic study of fusarium wilt resistant in white pea was done by Gautami, and she screened a number of genotypes from three pea species, that is Hortense, Elatius, and Album. She could not get flowering in Album, so she used Elatius to cross this cultivated garden pea, and she developed this a population of F1 back crosses and F2, and further these line were screened, and on the basis of screening data, they found that the single dominant gene was responsible for resistant to fusarium wilt in Pisum sativum subspecies Elatius. The study on heterosis for quality tests in cauliflower was done by Geeta Karigar, and she crossed a number of lines and tester in two different cauliflower groups, that is early and mid group. From these crosses, they have selected best combiner for yield and different nutritional quality of the cauliflower. And they also study the glucosinolate content, different types of glucosinolate in different group of cauliflower. So they found that high level of glucosinolate, especially glucoerosin, in case of early, mid, late, and late group of the cauliflower. Finally, they also gone for expression analysis of the glucosinolate pathway genes, and they found that the lines which are having high level of glucosinolate were also having the higher level of expression of glucosinolate pathway genes. Genome-wide association study for tomato leaf curl virus resistant in cucumber was done by Mr. Malthes, and he screened 100 genotypes, and from that he identified DC61, DC70, and DC91 as the resistant genotypes for tomato leaf curl virus on the basis of vulnerability index. And later on, based on phenotyping at different intervals and GBS analysis, Manhattan plot showed that chromosome number 3 and 5 are possible source of QTL for resistance against TOLCB and D. 400 genomic SSR primers from Cucumis sativus was screened for genetic diversity analysis based on molecular markers. The PIC value was recorded from 0.22 to 0.75. Identification of host involved in recovery phenomena post-leaf curl disease infection in chili was studied by Pallavi. Chili resistant line DLS selection 10 shows the recovery phenomena after appearance of symptom. So try to know the reasons behind this. To know these different transcription factors like aggregated protein, dicer-like protein, and RNA-dependent RNA polymerase, they were studied and their expression were studied in these genotypes. And they found that these different genes which are involved in the innate defense mechanism like MIB and Burki were highly transcribed, means their transcription level was very high in resistant genotypes and their transcription was very low in case of susceptible genotypes. On the basis of that, they found that finally that chili leaf curl virus and chili leaf curl Jadepur virus were the predominant viruses found in the high conditions, and they also noticed that the Burki gene and MIB gene were the mainly important for showing the resistance to chili leaf curl, and in the recovery mechanism their role was important. However, the role of RNA interference was not found in this, which is very common in other crops like tomato, etc. Physiobiochemical traits association and gene expression study for marketable bulb for methane Kharikonian was done by Mr. Jayant. He analyzed the lines for a number of morphological biochemical traits, and he found that a lot of variation for morphological traits, especially the leaf related traits, leaf length, leaf width, leaf area, was there was a significant difference after 60 days of planting in onion. The expression of flowering locus T genes, which is associated with the flowering initiation and bulb formation in onion, were also studied in different stages at 115 days after planting, and different group of the genotype, that is poor, medium, and good yielding. And it was found that the expression of the most of the genes were low in poor yielding genotypes, and the expression of the ACFT1 and ACFT2 gene was high in PKO1951, and expression of ACFT6 gene was found very high in leaves of the medium and high yielding genotypes. Assessment of genetic diversity and functional quality in garlic was studied by P. Karisman, and C used 29 Ilium sativum and 3 outgroup garlics for this study, and they could identify the different genotypes for different yield and quality traits. And based on the molecular analysis of 214 SSR markers, the genotypes were grouped into 4 distinct clusters. The normal garlic cloves were kept in oven for a longer period at 60 degrees Celsius temperature to produce black garlic, and it was found that after 40 days of curing, the nutritional quality and functional quality of garlic cloves were found best for storage of longer duration, and also the taste, etc. were also found desirable. Assessment of combining ability in heterosis for yield and quality traits utilizing petrolite CMS inbred in tropical carrot was studied by Mr. Janardhan Reddy, and he developed habits by crossing 4 lines and 12 tester. And on the evaluation trial, he found that hybrid DCatch 531 recorded maximum TSH number sugar and fructose, while DCatch 9837 recorded high carotenoid and lycopene content. And on the basis of yield performance, DCatch at 9848 recorded heterosis for root yield over bit parent, better parent, even over the check parent by 43%. Studies on physiological traits and QPL mapping of extended shelf life in cucumber were done by Suresh Paila. He screened a number of genotypes and found that GC48 was highly suitable for retaining the green color for longer period, and it has also got firmness, and there are also association was found that lower moisture loss and high photosynthetic rate were generally found in those genotypes which retained the green color for longer period. They also identified the 2 QTLs which were related to extended shelf life in cucumber at chromosome number 4. Genetic diversity and population structure analysis in bottle gourd using morphological and molecular markers were studied by Samantha. She found a very high degree of variability in case of leaf shape and size, fruit color, shape and size, and in molecular study with 100 SSR marker, she could group these 82 genotypes in 6 different clusters. Inheritance and identification of molecular marker linked to gynosis and its maintenance in bitter gourd was studied by Swamini Bhoi, and they studied the multiplication of these gynoecious lines in bitter gourd, and they found that in the genetics that single recessive gene regulates gynocism in, and single dominant gene controls discontinuation fruit rich pattern in bitter gourd. And they also found that the silver nitrate 250 ppm was found very effective for induction of male flower in gynoecious line. They also studied this tissue culture method for this multiplication and proper root induction or food multiplication. And they also identified this one marker which was linked with gynoecious and monoecious, which can distinguish both the parent, and that was a dominant type of marker. Optimization of sowing time and nitrogen level for seed productivity and quality in garden pea was studied by Mr. Bharat, and he found that the fourth number sowing gave the highest seed yield of 21.5 quintal per hectare in garden pea, and another finding of his study was 50% nitrogen and seed treatment with rhizobium foliar spray of urea at 2 times gave the 1.5 benefit cost ratio in USA 3 variety of this garden pea, which is early maturing type variety. The faculty and student of Division of Vegetable Science also received several awards and honors during this period, and students published very good publications, especially in the very high rated journals like Scientia Horticultural, Frontier in Plant Sciences, Journal of Horticultural Sciences and Biotechnology, Journal of Food Science and Technology, Genetic Resource, and Crop Evolution. Our faculty also participated during this period in international conferences in different parts of the country through virtual mode or some on the offline mode wherever it was possible. And some scientists also got training during this period on various aspects for upliftment of their knowledge. Thank you all. I acknowledge the director, joint directors, associate dean, head division of vegetable science, faculty and students for providing help and support for fulfilling these objectives during this period. Thank you very much.
Thank you, Professor Yadav, for your very good presentation. Now we have covered this School of Horticultural Sciences. So I will request our chairman, sir, for your kind remarks and comments.
Is it okay, sir, that I'm able to, you are able to hear me?
Yes, yes, sir. Yes, sir. Yes.
It has been my pleasure to hear the presenters of various students' research work by Dr. Srivastav, Dr. Yadav, Dr. Singh, and Dr. Ramasre. My pleasure to congratulate all the students who have been awarded or who are going to be awarded with the MSc and the PhD thesis. Naturally, the research work is of very high order. I have few simple rather queries related to, I mean, not directed to the students or the, but for my curiosity. What is the mechanism to coordinate entire activities? I mean, the students who are at IHR Bangalore and the Jharkhand, their research programs and the course programs are all coordinated at the IRI Delhi level, or they are fully independent? I think Madam Rasmi Agrawal will.
Yes, sir. Yes, sir. So they are actually they are monitored by IRI head office, but their problems are based on the local problems. They choose the problems which to cater to the local needs, and if their the eligibility, depending on the eligibility of the faculty to become guide, the guide is either from IRI like Jharkhand or outreach programs, or if their the faculty is not eligible to be the guide, then the guide is from IRI from the same discipline.
And there the faculty be at IHR Bangalore.
Yeah. Okay. Like for IHR Bangalore and for CIAE Bhopal, we have the PhD candidates only, and there most of the faculty is eligible to guide the students.
But the course, course program is approved at your level.
Yes, sir. All programs, they come at our level. Okay. So we approve the ORWs, sir. They come through BOS of their disciplines. Board of Studies, they do after the advisory committee meetings and Board of Studies, that it comes to our level, sir.
Now, another simple clarification for me would be, a lot of students are doing I mean, about 70-80% work is related to QTLs and the markers related to the various phenotypes. How much of that is being directly put to, let us say, the ongoing breeding programs or the research programs, where the principal investigators are getting advantage of the student's thesis, or is it the student does the work and the material extra is left somewhere and there is no follow-up action? I mean, what is the mechanism to integrate the student's research work with the ongoing program so that we get a real advantage out of that thesis? I'm sure there must be some, or is it
Yes.
the student does it and goes and then another comes and starts another experiment afresh?
Whenever a student is given a problem, it is given by a supervisor or the chairman, and he has a particular plan of work, sir. Either as an in-house activity, in-house project, or the externally funded project.
Ma'am, that
Under that,
is
sir,
okay.
it is continuity is maintained, sir, in most of the cases.
It gets into the mainstream research program by the, I mean, the advisor or the,
Yes, sir.
It gets into there.
Yes, that is part of the mainstream work. So, Director, sir, can highlight more on that, sir.
Very obviously, so most of these research programs, whether they are of basic nature or strategic nature or applied nature or translational nature, they are part of ongoing mandated program of the institute. They are not different from that. So they are very much integrated. And many times a student goes and the second student comes and he takes, for example, if a student has ended up with mapping some QTLs, the second student comes, he takes a fine mapping of those QTLs. Next will come, we will try to clone those QTLs and do functional validation and develop markers so that those markers can be used for marker-assisted breeding for transferring these QTLs into breeding program. So it's a complete chain. In fact, I would say complete value chain. There is no discontinuity in that.
Now, in the fruit crops, when they are talking about the QTLs and the gene mapping, etc., that heterozygosity of the progeny doesn't matter, because normally in the annuals we have been told that we need RILs, and in the fruit crops, is it whatever crosses they make, it is perfectly okay.
Sir, with due permission, can I?
Yeah, please go ahead.
Actually, sir, that is the biggest problem with the perennial fruit trees. The development of inbreds or pure line is just impossible in case of perennial fruit trees. So even we are taking the advantage of heterozygosity present in the parents, and even we are getting the segregation in F1 generation. So for that purpose, we are only choosing those loci which are showing some sort of polymorphism between the parents and how it is segregating among the progeny population. And this is a very, means I can say we have initiated the work in case of mango because there is no other report available in case of mango as far as the QTL mapping is concerned, sir.
No, but how does it help the, for example, somewhere I heard MAS for fruit quality in mango. Now, fruit quality in mango fruit is much more easy to evaluate as such rather than going into those finer QTLs and the markers.
Actually, in case of mango, the problem is that the genetics of important horticulture traits is poorly understood, sir. So that is a kind of obstacle.
Fine, but what is happening in every crop for every trait, there are QTLs. I mean, if somebody does some experiment, it seems there is a routine kind of method and materials and method, and you end up with some QTLs. So,
sir, this is, yes, sir.
And if somebody else will do it, he will end up with some other QTLs. So my concern is perhaps we are missing the tree for the wood, or it is a over excessive utilization of QTLs and mappings because of the instrumentation and the availability of the bioinformatics information and the programs. And in that process, perhaps we are losing the ground, or I mean, simple things may get complicated. For example, the mango itself is an example. The breeding for quality or assessment of the quality phenotypically is much more easier than perhaps using the MAS for fruit quality in mango.
Sir, with due permission, may I reply, sir?
No, most welcome.
Sir, actually, whatever our students are working on Mango, it is a part of a mega project where our team, under the guidance of Dr. N.K. Singh, we have deciphered the entire genome of the Sheri, Neelam, and Amrapali. And whatever genomic resources have been developed, we are trying to utilize those resources for the genomic assisted breeding. And QTL is part of that, sir. Otherwise, what if fun to go for developing such a huge quantum of genomic resources in perennial fruit trees, which is really very difficult to generate, sir.
No, my simple question is how much it goes into, I mean, how does it facilitate the selection of the fruit quality traits?
Sir, it will take its own time, sir. And now we are at this point that we have identified some QTLs. We are in the process of going for fine mapping of the mapping population, and then we will be validating.
You will directly select a mango variety based on the phenotypic performance of the fruit itself.
Sir, because of the heterozygous nature, we cannot predict the segregation of the
population. I mean, I have the fruit in my hands and I'm eating it and I'm judging it. There's no question of predicting it. The tree is there in the field. Its fruits are there. So the assessment of the fruit quality is much more easier directly than taking the indirect route of perhaps, especially in the fruit.
Provided the desirable traits are available in the germplasm. For example, I'm quoting one thing, sir, anthracnose, which is the biggest problem, but there is no genetic resource.
I'm not talking about anthracnose. My moot question relates to fruit quality. The mango fruit is in my hand. Can I judge it or not appropriately rather than getting the help of MAS?
No, sir, we can judge it. Phenotypically, we can judge it, but knowing how those traits are expressed, we can further modify that when using the latest genomic tools available with us.
Fine. Now, another simple question is the most of the thesis experiments related to post-harvest technology. There are various treatments. You are getting the shelf-life increased, quality is increased, various characters are modified here and there. How much of that recommended treatment can be practiced at the household level? Or is it only for the industries and the food processing industries? Or some recommendations which can be practiced at the household level? And that is very important because after all, 80-90% of the product is consumed at the household level. And that is across the, I mean, it is only applicable for the fruits as well as the vegetables.
Hello, Professor from Post-Harvest Technology.
Please.
Sir, can I respond, sir?
Yeah. Please.
Hello.
Yes.
That's a very valid question, sir. During your IRC, you have also iterated the same thing over there. The out of your finding, who is the ultimate takers. So the study we have here conducted 12 studies in post-harvest technology division. So 6 on PhD program and 6 on MSc program. Out of 4, they are related directly to application of some elicitors or you can say treatment for enhancing the shelf life and quality of 2 fruits and 2 vegetables. One is the mango, another is this.
Some detail is not needed. How is there any recommendation out of the various treatments related to enhancing the shelf life of the fruits or the vegetable, which can be practiced at the household level?
Definitely, sir. There is. For
example,
suppose that this application of different treatment like some of the in Kino Mandarin, application of hot water treatment or putting in them some perforated films, and that can enhance the shelf life. As in case of Kino Mandarin, putting this application of lemongrass oil and in perforator film of 200 SDP that have extend the shelf life for 6 days in house at cold level at room temperature in comparison to others. So that may be some of the, you can say.
Okay, fine. Now, another comment is related to polyploidy in mango, Dr. Srivastav. Polyploidy in fruit crops. I mean, polyploidy has never given any result even in the annuals. So how do you expect that polyploidy will help in the perennials?
Sir, with due permission, may I reply?
Yes, yes.
Sir, actually, we had two programs, two projects. One is to reduce the number of seeds in case of Mosami, which is highly seeded, and at the same time to develop seedless kinno. So for getting seedless one, this triplet breeding approach was preferred, and it is a part what student did.
Just to cut short the story, is there any example globally where the polyploid fruits are under commercial cultivation?
Sir, this watermelon.
Not necessarily occurring polyploids, induced polyploids artificially.
Sir, in fruit crops, this grapes, there are examples, sir. And even in citrus also from California, there are few reports that they have attempted this triplet.
Any product which has been induced as a induced polyploid and which is under commercial cultivation.
No, sir, I'm talking about the designed one. I'm talking about the targeted which were artificially created by crossing tetraplats with the diplats.
True polyploidy, is there any product in the fruit, across the fruit, which is under commercial cultivation anywhere?
Pardon, sir, I could not listen you.
Any induced polyploid in fruit crops under commercial cultivation anywhere?
Sir, in case of grape in Spain, they are using polyploids. So many polyploids of grapes are available, which are commercially cultivated in for wine grapes.
So it is better to use in the grapes rather than in perhaps the Guava.
Actually, sir, for getting seedless type of kinno and Mosami, either we have to go for mute or we have to go by this ploidy breeding. So here,
the ploidy is okay, please go ahead. But my concern was perhaps the polyploidy has not delivered that much as what was expected out of polyploidy, even in the annuals, etc. But still, if you feel that it is worthwhile, please go ahead. So I think my questions have been answered. I'm very happy to see that.
Except if you permit, I can submit something more.
Time
only 1 minute. Only 1 minute. Sir, one of my students, she have worked on irradiation with UVC application, physical application. Sir,
but I don't know. My concern था now since you have initiated.
आधा मिनट मेरे को दें, sir, please. So that I have communicated to APIDA.
Dr. Ramasre, first listen to what the Honorable Chair is saying. Then you come in for answering the question.
You have selected bitter gourd and the perhaps for the post-harvest increasing shelf life. To my knowledge, the most susceptible crop or the crop which is notorious for shelf life in vegetable is tomato and the Indian that muskmelon. But for the bitter gourd and the bottle gourd and cooker beets, etc., the reasonable shelf life is very much there. People are more concerned about tomato, which gets spoiled during transportation and the storage. And same is true about that indigenous or the Indian muskmelon, which was very juicy, etc., and which is just now has disappeared. So if something can be done to increase the shelf life of those crops which are prone to post-harvest losses, that would be a better option than doing the post-harvest studies in bitter gourd and the pointed gourd. Where the fruits are not damaged that easily. Bitter, fridge में मैं अगर bitter gourd रख दूँ 15 days, 20 days आराम से रहता है. No issue. Tomato क्यों नहीं लिया गया? Which is the flat tomato, that acidic tomato in South India, which they use in the summer and where the shelf life is very much, very less as compared to that square round tomato.
Now, Dr. Ramasre, if you have a specific answer, then please come in and tell. The question is very specific.
Sure. tomato for tomato I have not conducted, but for
Right.
that bitter gourd I have communicated the result to APIDA, and
No,
APIDA
bitter gourd
people,
you
they have
presented, that is
given
no.
me assurance that if you can upscale this technology, I'll give you full
While selecting the crop,
full-fledged.
While selecting the crop and the problem, a lot of inputs should go at that level, with the joint efforts of the faculty and the other colleagues, that what is the crop, what is the project, where the inputs, where the lot of efforts have to go. Selecting any crop XYZ and doing anything, I mean, that should be avoided. The problem it should be a burning problem at the field level. And then we must take the help of the students and the faculty members to chalk out the program so that we come out with a meaningful result, and which helps the farmers and the consumers at a larger scale. That was my only submission. The rest is absolutely fine. So from my side, sir, it is Dr. A.K. Singh, it is perfectly okay. It is my turn to thank you very much that I got a chance to interact with the faculty members, to hear what is going on. Naturally, that has helped me to refresh my memory. And thanks to Madam Dean PG and the 4 presenters and the advisors of the students who have done a fantastic job in terms of the MSc and the PhD thesis work. And I'm sure they are publishing the research. But is there any, I mean, I think publishing the paper is not a prerequisite for submitting a thesis, isn't it?
Yes, it is there.
It is a requisite in IRI, and yeah, so for PhD, at least 2 papers.
So
One, yeah.
So that is very important. Then that we, I mean, from my side,
MSc also. Yes. Right.
Okay, because just submitting the paper and writing, and before submitting the thesis, that's a good prerequisite for the student. Otherwise, they forget about whatever they have done. So nothing from my side.
Thank you, sir. Thank you so much for your kind input, and we have taken note of all the suggestions that you have given. You have asked very relevant questions. But the colleagues have not given relevant answer, as a matter of fact. And colleagues need to read more. And I am sure there are relevant answers available. Why it is being done, but colleagues need to, study that in deep, and then, probably you would like to communicate in writing how the genomic resources can help in improving the quality in perennials. For example, in case of apple, you have browning problem. You cut the apple, and after cutting some time, you get browning. That's a major problem, And if you buy genomic resources, if you identify the gene that is responsible for browning, because of which the oxidation happens and then phenolics develops, and if you knock down that gene, and that gene can be knocked down by genome editing now very easily.
Yes.
And then you get an apple, which you cut it and keep it, it will remain fresh for long, like it has been done in case of mushroom. Browning in mushroom was a major problem. And now you have got non-browning mushroom. The lifespan has been enhanced more than a week. Otherwise, it will turn, if you leave outside, mushroom will turn black and brown in color, and quality goes down. It becomes poisonous also. So there are applications, specific applications, and colleagues should read in deep and then try to emulate those points. Thank you very much, sir, for your kind time and interventions. And we are always encouraged with the presence of doyens in the respective fields like you. Thank you so much.
Thank you very much.
Dr. Ram. Thank you, sir. Thank you very much.
Yes, sir. Thank you, sir.
Critical input, sir. Yeah.
Thank you. So I am grateful to Dr. Harihar Ramji, former head vegetable science, GB Pant, University of Agriculture and Technology, and very senior renowned plant vegetable breeder, for agreeing to chair this session and blessing all of us. Sir, your vast experience is reflected in suggestion and comments. It will be very helpful to our faculty as well as our students in deciding the future course of research. So thank you very much, sir. Thank you. and presence of scientists of huge stature inspired us one and all, sir. Thank you.
Thank you so much.
Thank you, sir.
Thank you, sir. So
next.
we'll proceed to last session of today's celebration, and it's session 6th. And good evening once again to all of you. And we are having the session on the School of Social Sciences. So once again, I welcome each one of you, and so thankful that still we are more than 457 are there, but slowly we have gone down in a number of attendees. So with this, I once again welcome all of you on this presentation of the School of Social Sciences. And School of Social Sciences total has 30 thesis presentations. So this Social Sciences has 5 disciplines: agricultural economics, agricultural extension, agricultural statistics, bioinformatics, and computer applications. And total are 50 theses. Sorry, I by mistake I said 30. There are 50 theses, out of which 31 are MSc and 19 are PhD theses. And all those students whose significant achievements will be presented by the professors of these disciplines, they will be getting degrees during this 60th Convocation Week program. So friends, I'm very happy to say that we have with us our expert who will be chairing this session, Professor Rakesh Singh, Professor and Head Economics Division from Banaras Hindu University. So we are very delighted and very happy to welcome you. Today also you gave us a lot of your precious time, and we are really very delighted that you agreed to chair this particular session. And I'm also very happy to welcome our director. He throughout day, he had been really there to help us in these all deliberations, and all the heads and professors, students. And I must see that most of the students must have joined because they are waiting for the results also. For 2 days, their merit medal and the best student of the year medals declaration of the results. So with these words, I would like to now invite our director to kindly introduce the chairperson of today's session, this last session.
Thank you, Dr. Rasmi. We had Professor Rakesh Singh for the last 2 days as a member of the jury for selecting the IRI medal and Best Students Medal awardees. And as you all know, that Dr. Singh is an eminent economist at Banaras Hindu University. He's a graduate and postgraduate PhD from Pantnagar, and also worked at Indian Institute of Management. And very recently, those who participated in Agricultural Science Congress would know that Dr. Singh was the organizing secretary of the Agriculture Science Congress in Banaras Hindu University, and it was managed so well that everybody had appreciation for his efforts single-handedly. He put in a lot of efforts to organize this Congress very successfully. I mentioned the day before yesterday also that his understanding of the agricultural reforms in terms of farm bills is remarkable, and he was the most sought after resource person on most television channel when this agitation was going on. And that's where I heard many times Dr. Rakesh Singh on television. I had not known him before that. So that was the time when I got introduced to him, and I called him on phone that there are things where we have difference of opinion, and I would like to discuss at length with you sometime. And we did discuss on certain issues. So thanks, Professor Rakesh Singh, once again for accepting to our request and being here. And since the number of presentations are too many, actually, number of theses, as you said, I would request, in fact, the professors to do some kind of exercise where rather than going, individual thesis by presentation, if we can do some kind of grouping, if possible, wherever it is possible, where there is a common areas, and then we bring out the significant research findings. maybe putting together more than one thesis, the key findings can be highlighted. So with that, I would like to welcome once again each one of you. And over to Dr. Datta for the next proceedings.
Yes, sir. Thank you very much, sir. Thank you. We have reached to the last school, but not least. This is also equally important as other school we have, because social scientists add value to the agricultural research, what we are doing, and helps in socio-economic development of the farming community in India. So there are 5 disciplines we have under this school. So with the kind permission of Chairman Sir, may we take up the first presentation?
Please go ahead.
Okay, sir, thank you. So first presentation will be from Agriculture Economics by Professor Alka Singh.
Very good evening and welcome you all. On behalf of Division of Agricultural Economics, this is my proud privilege to present before you the significant educational achievements in the discipline. Sir, currently we have 48 students on roll, out of which 4 MSc and 5 PhD students are graduating this year. We have overall 17 faculty, including faculty from NIA, and out of which 11 are research guides. Sir, I am proud to say that this year student publications have increased to 27, and out of which more than 50% are in more than 6 NAS rating. Even 3 publications are having more than 10 NAS rating. These are the graduating MSc and PhD students along with the names of their chairpersons. Their research work has been categorized under 3 broader research themes. One is technology, resource and environment. Second theme is agricultural markets and value chain. Third theme is institutional innovations and rural livelihood. A study on employment diversification based on PLFS 2017-18 unit level data explored emerging trends of rural labor market and also assessed the impact of participation of non-farm activities on household consumption expenditure and poverty reduction. 2 things came out very clearly from this slide. Number 1 is much lower female participation rates in workforce as compared to male, and the other thing is that there is significant decline in rural female in labor force participation rates in the later period, that is 2017-18 as compared to 2011-12. Though bulk of workforce is still employed in the farm sector, however, increasingly employment diversification towards non-farm sector, particularly in construction and services, is clearly visible for rural male and services for rural female. The impact of participation in non-farm sector on household consumption expenditure and poverty was estimated for northeastern states using inverse probability weighted regression adjustment model. The ATET estimates clearly showed a significant increase in consumption expenditure by the households whose principal earning is from non-farm activities as compared to those whose earning is coming from the farm sector. The similar results were also visible once the impact of occupational diversification was estimated on poverty. The study also assessed rural youth perceptions and aspirations about farm and non-farm sector in selected villages of Arunachal Pradesh based on 200 rural youth of 18 to 40 years of age categories. The study clearly spelled out that female youth have relatively positive perceptions and higher aspirations than male youth regarding farming. Hence concluded that there is need to transmit information and educate the youth regarding the scope of farming and agribusiness opportunities in the region, along with developing and strengthening infrastructure and higher exposure towards training. Processing industry is recognized as one of the sunrise sector and has considerable growth potential for employment to rural people, as majority of them are based in rural India. However, this industry in India is characterized by small, that is micro, small and medium units. Only 0.84% of the firms have R&D units and 1.09% firms received foreign capital. Though the market size of the industry is estimated as 8151 billion rupees at 2011-12 prices, however, the industry is still dominated by traditional sectors such as grain, starch products, dairy, oil, fats, and sugar, which contributes more than 2/3 of the market. Productivity analysis of the industry clearly showed that the industry is largely being driven by the input growth followed by TFP growth. The technological gains are more visible only after 2004 and the estimate of the employment function clearly shows that fish, meat, and animal feed has higher employment potential as compared to the traditional sectors like sugar, grain, and dairy. Productivity spillover effects of foreign direct investment was also analyzed by the random effect panel data regression model, and results clearly indicated a positive relationship between productivity and FDI. However, R&D intensity in the industry is negligible, hence incentivization of investing in R&D is a need of the hour. Study examined incidence and magnitude of borrowing as well as pattern of farm investments in agriculture households of semi-arid tropic region of India based on VDSA data of 2009 and 14. The study revealed that more than 80% of households borrowed from different sources, mainly for the purpose of for the agriculture related social functions, repaying old debts, major repairs, etc. However, the silver lining is that formal sources borrowing has increased over a period of time, but farm households' investments in non-farm assets increased during the period. The study on performance of custom hiring centers in Rayalaseema region of Andhra Pradesh examined the accessibility and adoption of improved farm machineries across the farm size categories, as well as also assessed changes in cost of operations, profitability, and efficiency in crop production due to adoption of improved farm machineries owing to CHCs. The study highlighted that there is a higher mechanization index for CHC farmers than non-CHC farmers. Also, share of machine labor to total cost is high in case of non-CHC farmers compared to CHC farmers. The study estimated that custom hiring centers affiliated farmers were found to be more technically efficient. The study also underlies the need to improve access of farm machineries to small and marginal farmers through establishing more number of CHCs and providing sufficient number of implements to custom hiring centers for improving services on time. This study analyzed demand and supply of irrigation water in eastern Yamuna Canal Command area of Uttar Pradesh, also examined farm-size groundwater energy productivity relationships in crop production in western Uttar Pradesh, and developed scenarios for sustainable and inclusive use of water resources. The estimation of water balance in different agroclimatic zones of Uttar Pradesh clearly indicates that mid-western plains and western plains zones are highly water deficit. The cost of extraction of groundwater by electric operated wells as well as diesel operated wells under different farm size groups in the study area was estimated, and the results shows that cost of groundwater extraction through electric operated tube wells are much lower as compared to the diesel operated pumps. The analysis of irrigation cost per hour also shows that it is lower in case of flat tariff for the farmers. Ecosystem services delivered by Eastern Yamuna Canal Command area was estimated based on the current market price, and it was valued at 1123 million rupees, which is much higher than the total working expenses of the command area. This piece of PhD study addressed research questions as benefits of solar installed pumps over electric and diesel pumps, and also identified factors influencing the adoption of solar pump technology in two states of Bihar and Rajasthan. The results revealed increased cropping intensity over SIP farms than non-SIP users, also reduction in cost of irrigation, undisrupted water supply during the critical stages of irrigation, also lead to yield advantage of 5 to 15% across various crops. They were also found to have higher technically efficient. Regarding incremental social benefits, because of the high subsidy, the ISPs of SIPs over electric pumps comes out to be negative in both the states. However, the SIPs have positive incremental social benefits over diesel pumps in Rajasthan. The undulated topography and distance from the market in case of Rajasthan, farm fragmentations in case of Bihar, and theft in both the states were found as significant constraints in adoption of SIPs. Access to credit and training were found to have significantly pushing up the adoption process. Sustainable soil health management is a critical challenge in the country, and soils in many parts of the country suffer from widespread multiple nutritional deficiencies. This is a major constraint in improving crop yields and realizing full potential of agriculture. This study constructed district-wise mapping of macro and micronutrients deficiency based on Soil Health Card MIS database, which is available in public domain now. Also constructed composite soil fertility indices with and also examined its association with paddy productivity in Andhra Pradesh. Based on the CSFI indices, districts of East and West Godavari and Guntur had highest score of CSFI, which is indicating better soil health status in the state. This also analyzed and showed that significant impact of CSFI on yield of paddy in Andhra Pradesh. Based on survey of 180 sample farmers of Nellore and Chittoor districts of Andhra Pradesh, the study assessed impact of soil health cards on nutrient use and productivity. The study showed that significant decline in the use of nitrogen and phosphorus nutrients and also increase in micronutrient fertilizer application on SHC farms as compared to non-SHC. The study concluded that membership to farmers organizations and trainings emerged as significant and positively influencers of SHC adoption in the study area. The study highlighted that sample collection has to be made properly and in the presence of farmers so that a trust can be built, as many sample farmers felt that the sample is not from their field. Regular soil health card trainings along with field demonstration with the active participation of the farmers would help in effective adoption of soil health card in the study area. The study on production risk in major pulse crops examined the profitability, sources of growth, and instability in production of selected pulse crops. Also identified the factors associated with risk reduction in pulse crop production. The results revealed that growth in gram production is driven by both area and yield, whereas growth in arhar production is driven by mainly yield. Analysis of profitability clearly indicates very low net profit or even negative in few cases. Just and co-production function estimates revealed that fertilizer, irrigation, seed and mechanization contributed to reducing risk in case of gram, whereas in case of arhar, labor, seed, fertilizer and irrigation contributed to reducing risk in arhar production. Import of edible oil accounts for 50% of India's agricultural import bill. This piece of work identified trends in import of edible oils, assessed the price volatility of selected edible oils in domestic and international markets, and also estimated import demand functions and their elasticity. This piece of work clearly showed that domestic prices of edible oils are affected by the changes in international prices during the study period. The estimates of import demand function of palm and soybean oil clearly showed that their imports are positively affected by the growth in income of the country as well as negatively impacted by the domestic production. Hence, policy initiatives to boost domestic production is the need of the hour. Palm oil and soybean oil alone constitute more than 80% of import basket of edible oil, so policy interventions to be created in near future should be based on the import responsiveness of these two oils. Sir, this year we have also conducted 3 short-term need-based online training programs for PG students of social science discipline and also uploaded video lectures on website and social media. Sir, I am proud to share this slide as our students have increasingly published in multidisciplinary high-rated NAS journals. Recognition of students' research at various national and international forums. These are faculty awards and honors. In the last but not the least, I thank all out support of Director IRI, Dean and Joint Director Education, and also acknowledge the contributions of all students, faculty and staff of the division in carrying out various academic activities in the division. Thank you. Thanks to one and all.
Thank you, Professor Alka Singh. for very nice presentation. And now, Chairman Sir, if you kindly permit, we will go for second presentation, and at the end we will have comment and remarks.
Yes, sir. Sure, sir. Please.
Okay, sir. Thank you, sir. So next presentation will be by Professor Ravindranath Padaria on behalf of Agricultural Extension.
Honorable Chairperson, respected Director, Dean and Joint Director Education, dignitaries, ladies and gentlemen. On behalf of the faculty and students of the Division of Agriculture Extension, I extend a very warm welcome and greeting to you all. There are 21 faculty, out of which 13 are research guides. Last year, Dr. Sukanya Barwa was inducted as research guide for master's program. We have one Reverend Adjunct faculty, Dr. P. Das, who had been the Deputy Director General of ICR. We offer 23 courses in 2 semesters, which include Scientific and Technical Writing, Research Ethics, Disaster Management, Science and Society. In this convocation, 9 students will receive degrees, out of which 3 are from PhD and 6 are from master's program. The major thrust research areas of the division had been development of extension models and strategies, entrepreneurship development, farmer-led innovations, gender empowerment, and capacity building. Inspired by Gandhian observation that India lives in villages, and if villages perish, India will perish too, Sangeeta Bhattacharya took up a study on socio-economic impact of Sansad Adarsh Gram Yojana in Maharashtra and Telangana. Her idea was to make a comparison of Sansad Gram as well as the model villages developed by philanthropic or collective actions of the farmers themselves. In order to make a comparative analysis of Sansad Gram Yojana villages as well as the model villages, she identified 7 indicators which included health, education, infrastructure, ecology, women empowerment, farm and livestock. She found that in Maharashtra, Sansad Gram Yojana villages scored high in terms of health and farming, while in Telangana, education, farming, livestock and ecological status were higher in Sansad Gram Yojana villages in comparison to model villages. Government public liaison, infrastructure and education, local political environment, collective power of villagers were identified as the facilitators for development of model villages. She suggested for ensuring convergence and dovetailing of schemes for effectiveness of Sansad Gram Yojana. The Government of India heralded an optimistic pathways for development of the lesser developed districts in form of aspirational district program. Arun Kumar took up a study to make an assessment of financial inclusion and skill development in agricultural sector through aspirational district program. The study was carried out in Karnataka. He found that the extent of financial inclusion was in the range of moderate degree. Agricultural laborers had lower extent of financial inclusion. Though majority of the respondents had high level of awareness about financial products, the level of use was low, while the majority of the respondents stated that access to financial products was difficult. As a result, level of satisfaction was in the medium range. The Government of India has laid strong emphasis on promotion of farmer-producer organizations. 10,000 farmer-producer organizations have to be developed. In this regard, Ankit Mahapatra conducted a study on FPO-led extension approach for organic turmeric farming in Kandhamal district of Odisha. He identified the factors which contributed to the success of one of the FPOs known as Kasam in Kandhamal: better price for produce compared to local traders, good infrastructure, facilities, assistance in availing the benefits of various government schemes were the major factors. There is increase in use of social media for extension service delivery. Adupa carried out an analysis of perception and extent of uses of social media among extension agents in Andhra Pradesh. The study was carried out in the districts of Srikakulam, Guntur and Chittoor. 160 extension agents and 63 farmers comprised the sample size. She found that around 29% of the extension agents had unfavorable perception towards social media, while 20% had favorable perception towards social media. Orientation to traditional dissemination methods, lack of technical knowledge and skills, lack of credible quality information, and a large number of other sources prevailing were the major constraints for use of social media by the extension agents. Skill India is one of the major campaigns of our nation. Harshita carried out a multidimensional study of agricultural skills among rural youth. The study was carried out in Hassan and Chikmagalur districts of Karnataka with sample size of 120 rural youth. She found that a majority of the rural youth had low to moderately favorable attitude towards agro enterprises. She identified that achievement motivation, risk orientation, self-confidence, and hope of success were the major factors which determined the attitude of rural youth towards agro enterprises. Atmanirbhar Bharat is another major campaign of our country. In order to attain the objective, we have to promote entrepreneurship development. In this regard, ICR has developed several agribusiness incubation centers at institute as well as universities. Sagar took up a multidimensional study on agribusiness incubations under Business Planning and Development Unit of IIHR, Bengaluru. It was found that a majority of incubatees had low to medium degree of entrepreneurial behavior. Scientific orientation, closeness with support system, rational orientation, self-reliance, and media utilization were the major factors which explained their entrepreneurial behavior. The extension agencies, while promoting entrepreneurship, must take these factors into consideration. Mr. Sushil Kumar took up an analytical study of effectiveness of agribusiness incubators of IRI, CFET Ludhiana, NARM Hyderabad, and RUCITY. 60% of the business planning development trainees found that the content of various entrepreneurship development training programs had medium utility, relevancy, and interest. A majority of the farm trainees had settled with the help of bank finance. Therefore, he suggested that more emphasis should be laid upon curriculum development for the various entrepreneurship development program and provision of finance to the rural youth for taking up agribusiness. Seed production could be one of the potential agribusiness for the farmers. Felster took up a study on seed source network with respect to wheat and green gram. The study was carried out in Haryana as well as Uttar Pradesh. She observed that the farmers in Haryana explored more of formal seed sources in comparison to informal sources, while in Uttar Pradesh, farmers utilized more of the private shops and other informal sources for procurement of seed. Therefore, promotion of seed production should consider the farmer preferences of seed sources while taking up seed production enterprise. She also evaluated the farmer's preference for the traits and qualities of two crops, wheat and green gram. For wheat, she found that farmers preferred more of yield, market rate, dwarf stature, and lesser incidence of pest and diseases and less water requirement. While in the case of green gram, the farmers preferred the traits of higher yield, bold and shiny grain quality, less incidence of pest and disease, synchronized maturity, high market demand, and big plot size. Alok carried out a study on stakeholders' risk-benefit perception and acceptance behavior of GM crops in India. He carried out his study in Maharashtra, Rajasthan, and Karnataka with a sample size of 610 respondents, which included 360 farmers, 160 urban consumers, 60 social activists, 30 scientists, and 10 traders. The major GM crops considered for the study was mustard, maize, and cotton. Dependency on seed agencies and incidence of secondary pests were the major risks perceived by the farmers. Yield and drought tolerance were the most preferred traits for the GM crops. They considered seed cost not so significant if the yield was assured. Farmers' acceptance or purchase intention was significantly affected by benefit perception as well as their attitude. An attempt was made to understand the risk communication about GM crops. In order to attain the objective, content analysis of the two dailies, that is the Hindu and the Indian Express, was done. We can find that during 2016 and 17, there were the maximum number of news articles related to GM crops. However, most of the articles had the negative frames rather than educative frames. Even the frequency of news articles evaluating field performance of GM crops or frequency of knowledge and awareness oriented articles were very less. It emphasizes that we have to have a communication approach to promote GM crops in India. The faculty and students of the division have made significant stride in publications. We had 15 publications of NAS rating greater than 6. We organized one online training on life skills and personality development for the technical staff. Our Honorable Director has made an innovative initiative for developing Pusa Samachar to communicate science among the stakeholders. Our faculty, Dr. R.R. Burman, Dr. Gurjesh Mehra, Dr. Sangeeta are playing key role in it. Our students have won several awards and honors. Mr. Ankit, Mr. Arun, and Dr. Sangeeta won Best Oral Presentation Award. We express our sincere gratitude to Director IRI, Dean and Joint Director Education, Associate Dean, Joint Director Research and Joint Director Extension for their insightful guidance. We are also thankful to PG School IRI for the various grants. I am also grateful to all the faculty and students of the division as well as farmers and respondents for their valuable contribution. I express my sincere thanks to all.
Okay, thank you, Professor R.N. Padaria, for your very nice presentation, that too within stipulated time. Thank you very much. Now we will move to agriculture statistics. Professor Srini Varghese will be presented.
Good evening, everyone. I am here to present the significant educational achievements in the discipline of agricultural statistics. I begin by quoting Rogers. Statistics and numbers are no good unless you have good people to analyze and then interpret their meaning and importance. I unquote. This graph depicts the number of students in agricultural statistics. Our first batch of MSc and PhD passed out in 1966 and 68 respectively. This year 14 are taking degree and there are 44 enrolled currently, 13 in MSc and 31 in PhD. Total number of courses offered in agriculture statistics is 44 with a total of 104 credits, which includes 84 in theory and 20 in practical. There is a total strength of 31 faculty in which 27 are faculty of agricultural statistics only and 4 are dual faculty in agricultural statistics and in bioinformatics. Out of 27 in statistics, 11 are eligible guides for both MSc and PhD, whereas 5 are only for MSc. And out of 4 in dual, 2 are eligible for both MSc and PhD. These are the 4 major areas of research. 5 students worked on design, 4 on statistical modeling, 4 on sample surveys, and 1 on statistical genetics and genomics. These are the 5 PhD students receiving degree this year, and these are the 9 MSc students receiving degree this year. First major area of research is designing and analysis of experiments. Sumit Saurav worked on the topic robust designs for bioequivalence trials. These trials are conducted to investigate the degree to which clinically important outcomes after receiving a new formulation resembles to those of a previously well-established formulation. An important area of application is the evaluation of veterinary medicinal products. Objective was to study the robustness of designs for bioequivalence trials under various disturbances or violations from assumptions. Due to scarcity in the availability of homogeneous, healthy subjects for experimentation, the same set of subjects may have to be used for repeated evaluation in these trials. Hence, the subject may exhibit time trend over periods, or there may be dependency in the observations. Also, few subjects may respond differently to the same set of formulations, making them outlying subjects. There may be outlying observations as well. This study gave out some series of robust and efficient designs in the presence of trend, autocorrelation, or outlying subjects or observations. These are some publications or conference presentations by the student. Constant block sum partially balanced incomplete block designs, where block sum with respect to treatments for all blocks remain constant, are gaining rapid momentum. In literature, trend-free block designs and constant block sum partially balanced incomplete block designs are studied separately. Hence, Satyam undertook the objective to obtain trend-resistant constant block sum PBIB designs. The study gave 3 classes of such designs, out of which 2 are affine resolvable class of PBIB designs. It also gave SAS macros and catalogs of these designs. This is a design generated through SAS macro. Here is an example of constant block sum PBIB design obtained through magic square. This is an affine resolvable block design. These designs are linear trend-free designs having constant block sum property. Here the constant block sum is 15 in each block. There are two associate PBIB designs with an underlying L2 association scheme. Our next MSc student Pramod worked on position balance block designs. In consumer experiments and sensory analysis, where panelists are required to rate number of products according to attributes such as sweetness, spiciness, or overall acceptability, rating given to a product may depend on whether it is being given at the beginning, middle, or at the end of the session. Hence, it is important to consider designs which are balanced for position effects. Also, there may be a requirement of incomplete block designs as they will reduce the consumer fatigues. Hence, the objective was to develop a method or algorithm for obtaining position balance or nearly position balance BIB designs. Outputs are algorithm for obtaining these designs, catalogs, and R codes. Here is an example of a design generated through the R code. These designs can be very efficiently used in FMCG product manufacturing industries or bakery or dairy product industries. Here comes an example. The original block design is not position balanced, but the algorithm converts it into a nearly position balanced design. Here, all the treatments are appearing at each position once or twice. Row column designs are useful for experimental situations in which there are two cross-classified sources of heterogeneity is present in the experimental material. In some experimental situations, due to cost or time constraints, experimenter may be interested in the estimation of main effects and some selected two-factor interactions only. Also, there may exist situation where there is a requirement of only 2 units in a row column setup. Kaushal worked on these aspects and obtained RCDs with 2 rows with specific two-factor interactions of interest and under fractional factorial structure. He has also prepared catalogs for these designs for n less than 10. These are examples for such situations. In the first example, two-factor interaction A into B is not estimable, and in the second example, all two-factor interactions are estimable along with the main effects. The relationship between numerous explanatory variables and one or more response variables is determined and quantified using response surface methodology, which is then utilized to optimize an underlying process. The treatment combination administered to an experimental plot may not only affect the response from that plot, but also from neighboring plots. In response surface studies, considering neighbor effects, it may not be realistic to assume same levels for all the factors. Hence, Ashutosh undertook this study with an objective to obtain methods of constructing designs with mixed levels of factors in the presence of neighbor effects for fitting response surfaces of second or higher orders. A response surface with S1 raised to N1 into S2 raised to N2 into S3 raised to N3 combinations has been considered. The developed designs are either rotatable or partially rotatable depending upon the model considered. R codes have been developed. A package called RSD-NE has been published at CRAN. This is a snapshot of the same. These designs are used in fields like agriculture, electronics, post-harvest processing, etc. Second important area of research is sampling techniques. Our PhD student Nitin considered the problem resampling methods of variance estimation for two-phase sampling. It is common in surveys to use two-phase sampling when it is relatively inexpensive to draw a large first-phase sample for which auxiliary variable alone is observed. A second-phase subsample of the initial first-phase sample is then drawn and both auxiliary and characteristic of interest are measured. Prediction approach is applied to predict the non-sampled units in surveys. In two-phase sampling, there are n-n non-sampled units, so it is required to develop an estimator based on prediction approach and variance estimation procedure for the same. An additional reason is the presence of non-response. The objectives were to develop robust estimators and its resampling variance estimation techniques and to study the impact of imputation on resampling variance estimation techniques for two-phase sampling in case of missing observations. The findings include robust estimators, 2 bootstrap methods of variance estimation of the proposed prediction-based estimator and validation of the results through simulation study and better performing ratio and regression methods of imputation techniques. These are some publications and conference presentations by the student. Multiple frames are preferably used when it is difficult to obtain a single sampling frame that covers the whole population, or two or more cheaper sampling frames are available from different sources covering the entire population. So our PhD student Rajiv dealt with dual frame surveys as a special case of multiple frame surveys, considering two frames covering the entire population and an independent sample is taken from two frames whose union covers the population. Also, sometimes it is tedious to obtain an unbiased variance estimator of the post-stratified estimator of population total under dual frame surveys. The objective was to develop resampling-based unbiased variance estimation procedures for the estimator of the population total under three different cases of dual frame surveys as given here. The proposed variance estimation procedures are almost unbiased. There is a reduction in percentage relative bias and relative stability of the methods due to the use of proposed rescaling factors, and the study resulted in a more dependable proportional stratified bootstrap method using regression imputation of missing observations. Some publications and conference presentations by the student are given here. Under stratified two-stage, two-phase sampling, strata are considered as mandals or talukas, first stage units as villages, and second stage units as CCE plots. Practical feasibility of CCE techniques for multiple picking crop like cotton is a matter of concern in the country. So, our MSc student Krishna took his research problem with an objective to develop methodology and perform empirical evaluation using real data for estimation of crop yield at district level using calibration approach under stratified two-stage, two-phase sampling. The findings include a methodology for estimation of crop yield at a district level using calibration approach, a separate regression type calibration estimator, and a combined regression type calibration estimator of the population total and population mean when size of all PSUs is known as well as when size of PSUs is unknown, approximate variance and estimate of variance of the developed estimators. If the developed approach works well for other crops also, this can be used for crop yield estimation under Pradhan Mantri Fasal Bima Yojana for obtaining more efficient crop yield estimators and optimizing the number of CCEs. General crop estimation surveys based on crop cutting experiments are conducted for estimation of crop yield following random sampling approach for almost all major crops. About 13 lakh CCEs are conducted every year under this scheme. Due to Pradhan Mantri Fasal Bima Yojana, which is yield based, this number has significantly increased to more than 1 crore. Now the government is stressing on the use of advanced technologies for crop yield estimation and for reducing the number of CCEs. So, our MSc student Naveen has undertaken his study with an objective to develop methodology and perform empirical evaluation using real data for estimation of crop yield at a district level using random forest spatial interpolation technique. Major research findings are crop yield estimation procedures using spatial random forest technique, including the spatial variables like distance and nearest neighbor as covariates. For prediction of yield at unknown locations, the estimates obtained using RFSI were found to be at par with kriging and better than inverse distance weighing. RFSI is a faster, flexible, and easy to use interpolation technique. It reduces the cost, operationally more convenient than the GCES procedure. Methodology can be advantageously used for crop yield estimation with reduced number of CCEs. This flowchart depicts the steps involved in the interpolation technique. Next important area of research is statistical modeling. Bayesian technique has a lot of advantages over the traditional ones. Time series model with exogenous variables has the capacity to identify the underlying patterns in time series data and to quantify the impact of environmental influences. To improve upon the existing models, our PhD student Yasin has framed his objectives under Bayesian framework to develop Sarima X model and to develop Gach X models. His major findings are these two superior models, Sarima X and Gach X. For planning a new scheme to develop the condition of Indian economy, the government needs a reliable forecast of various aspects. Time series analysis is very efficient to meet this need. Bayesian framework for Sarima-X and Gach-X models incorporating explanatory variables has a wide applicability here. These are some publications and conference presentation by the student. The feature selection algorithms assist in selecting only those features that are relevant in the predictive algorithms. Machine learning techniques have very few prior assumptions and are data-driven, and hence they provide great deal of flexibility for modeling and forecasting the crop yield. The major research gap was under regression framework using weather data, N-P problem will arise, and hence our MSc student Manoj has proceeded to evaluate the performance of crop yield forecasting models coupled with the feature selection in regression framework and to investigate the suitability of machine learning algorithms for crop yield forecasting. Major findings of the study are best feature selection algorithms like CBFS, Lasso, and random forest for prediction with a regression model. Support weighted regression was observed as the best machine learning technique. The feature selection with the regression framework was found to be more efficient as compared to the machine learning technique. These findings can play an important role in the agriculture domain with crop yield depending on multiple factors like land use, water management, and many weather parameters. This is a publication brought out by the student, and this is a conference presentation. In moving average model, equal weights are allotted to all the observations of the time series. In exponential smoothing, weights are assigned in exponential manner as recent observations getting more weights than the older ones. So it doesn't capture entire pattern in the time series, and the entire characteristics of the time series can't be explained properly. Hence, our MSc student Praveen has carried out his research work with the objectives to develop time-varying smoothing model using weights as a function of lagged observations, and also to frame hybrid model using machine learning techniques. His major findings include a hybrid time-varying smoothing model for forecasting agricultural prices. The residuals of selected time-varying smoothing model is fitted with time delay neural networks for predicting the non-linear pattern in the time series. The hybrid time-varying smoothing model achieved better forecasting than the normal existing smoothing models. In terms of price forecasting of non-seasonal crops, the proposed model will be more appropriate in short run. Volatility forecasting is an important area of time series where short and long-term volatility components influence the price series differently. The strength of Gach-Midas model has been barely explored for volatile agricultural series. Hence, our MSc student Anushka took the objective to investigate Gach-Midas model for forecasting agricultural price volatility and then to perform a comparative analysis of this model with the Gach model. She has written some R codes for the purpose. Price volatility can be forecasted efficiently for datasets with higher frequency when datasets with lower frequency are used as exogenous variables in Gach-Midas model. Accurately forecasting price volatility is very important for both policymakers and farming community. Next and last important area of research is statistical genetics and genomics. In the field of DNA sequencing, genotype by sequencing is meant to discover SNPs in order to perform genotyping studies such as JIVAs. A most commonly occurring problem in GBS is the presence of missing observations. Quite often, the standard statistical models may not be able to handle such missing data. Hence, in his PhD work, Srikanth has taken the objectives to identify a suitable imputation method against varying levels of missing marker data, to identify a robust genomic selection model against incomplete genotyping data, and to develop an efficient empirical procedure for estimation of breeding values in the presence of missing data. The study revealed that imputation based on random forest, expectation maximization, and singular value decomposition exhibited high imputation accuracies. Prediction accuracies of GS models based on machine learning algorithms outperformed Bayesian and Pulp approaches. RF imputation followed by RF regression based GS model showed highest accuracy in predicting breeding values. Now coming to the recognitions, 2 of our students were recognized for their good academics. Along with academics, our students are also very actively involved in co-curricular as well as extracurricular activities like quiz, painting, logo designing, poster presentation, sports activities in various events organized by IASRI. In the year 2021, our faculty has published 75 research papers. A summary distribution of the same is given here. Some faculty members were recognized for their work through awards. Some have participated in various international trainings, webinars, workshops, conferences, etc. virtually during the year. ISRI is known for organizing various types of training programs. Our faculty is involved in this aspect as well. Some are involved in teaching senior certificate courses also. Thank you.
Yeah, thank you very much, Professor Shini Bargis, for very nice presentation. Now, Professor Anil Rai will make presentation for bioinformatics.
Namaskar. I am Dr. Anil Rai, going to present significant educational achievement in the discipline of bioinformatics. In this convocation, total 8 students are going to take degree. 3 PhD students, that is Chiranjeev Sarkar, Bulbul Ahmad and Priyanka Guha Majumdar, and 6 MSc students, that is Vivek Shah, Laldhari Patel, Shivdharan S. Jhilri, Asif Ali V.K., Chandana V. Chiranjeev Sarkar worked under the guidance of Dr. Rajendra Prasad. The thesis title is A Study on Gene Regulatory Network for Rice Blotch Disease. Objectives are to construct a consensus gene regulatory network derived from various methods for rice blotch disease, validation of the consensus gene regulatory network, to identify the key genes in consensus network, development of web tool for gene regulatory network for rice blotch disease. In his study, gene expression data of rice blotch disease have been downloaded and a consensus gene regulatory network has been prepared and evaluated. It has been found that consensus gene regulatory network has more stability and accuracy than 4 individual methods which has been generally used in traditional analysis, that is correlation, PCR, PLS, and ridge regression. He also developed a web tool for gene regulatory network construction and published 2 papers out of his work. Next student is Bulbul Ahmed. He did PhD under the guidance of Dr. Anil Rai. The thesis title is Development of a Deep Learning Based Methodology for Functional Protein Classification. Objectives are to develop a deep learning based methodology for binary class classification of protein, to evaluate the performance of the developed methodology with existing deep learning and machine learning algorithm, to construct a web-based deep learning tool for classification of. In his study, he applied support vector machine, random forest technique, and a deep learning method which is called long-term short-term memory model for abiotic stress conditions. He also proposed a novel activation function, namely SILU, which has been implemented using this LSTM model, which has been developed under this study. He also developed a web-based server and tool for the classification of the protein and published. Next student is Priyanka Guha Majumdar, who worked under the guidance of Dr. A.R. Rao. Thesis title is Development of a Model for Classification and Characterization of RNA. Objectives are to develop a classifier to discriminate non-coding RNAs from coding RNAs, to categorize non-coding RNAs into different classes using machine learning approaches, to study the involvement of non-coding RNAs in gene regulatory mechanism of biotic and abiotic stress response, to develop an online prediction server. She applied coding RNA as a positive set and non-coding RNA as a negative set of 63 plant species. And for binary classification of plant coding and non-coding RNAs, she applied machine learning tools, that is random forest, support vector machine, artificial neural network, and a deep learning method that is called DNN, autoencoder. Feature-based DNN models were developed. And she found that binary classification random forest shows highest classification accuracy. She also developed web-based tool and published a paper from this work. Next student is Vivek Shah from MSc, who worked under the guidance of Dr. D.C. Misra. Thesis title is identification and characterization of long non-coding RNA in rice bean. Objectives are to identify novel long non-coding RNA in rice bean seed, to identify and annotate targets of predicted long non-coding RNAs, to design and develop a web resource for long non-coding RNA in rice bean seeds. He took a sample of 55,773 transcripts from rice bean seed sample, and he found that from the target annotation, it was observed that long non-coding RNA can regulate developmental process of rice bean seed through long non-coding miRNA interaction and miRNA-mRNA interaction mediated regulatory network. He also developed a web resource for the same. Next student is Lal Dhari Patel from MSc, worked under Mr. Sanjeev Kumar. Thesis title is Deep Learning for Predicting Breeding Value Using High Throughput Genotype and Phenotype Data. Objectives are to develop deep learning model for predicting breeding value for drought responsive trait in wheat. Second, to evaluate the performance of developed model for predicting breeding value for drought responsive trait in wheat. Next student is Siddharsan Srishal Jilri from MSc, who worked under the guidance of Dr. Manoj Grover. The thesis title is Identification and Characterization of BGIP and DUF gene family from developing seeds of Vigna umbellata. Objectives are to perform identification of BGIP and DUF transcription factor gene family involved in seed development, and to perform characterization of the BGIP and DUF transcription factor gene family. This is the workflow of the study, and the flow diagram is given in this slide. Next student is Asif Ali VK from MSc, who worked under the guidance of Dr. Anu Sharma. Thesis title is Phylogenetic Marker Gene-Based Approach for Binning of the Genomic Data. Objectives are to develop semi-supervised binning strategy for metagenomic data using phylogenetic markers gene, and to evaluate the performance of the developed approach. He computed RAND index and precision and recall and sell-out index. The basic finding of his research is that the visualization using BH-TSNE algorithm is highly promising for the TNF feature matrix, and the developed semi-supervised approach improved the binning results of the metagenomic. Next student is Chandana V from MSc. She worked under the guidance of Dr. S.B. Lal. The thesis title is Prediction of Enzymes Involved in Bioremediation Using Aquatic Metagenome. Objectives are to identify the enzymes involved in bioremediations of pollutant using aquatic metagenome, to compare the bioremedial enzymes generated from the places with various degrees of pollutant with respect to abundance, and to identify the related pathways of the enzymes, and to develop a web-based tool for searching bioremediating enzymes. She obtained bioremediating enzymes, organisms in which they are present and pollutant they degrade, such as dye, hydrocarbon, and plastic. And she also developed web-based search tool, which is beneficial for searching bioremediating enzymes online. In bioinformatics, total passed out students in MSc are 36, and from PhD 6 till now. Currently, we enrolled 10 MSc students and 23 PhD students. The distribution of these students across different years are given in this slide. We offered 39 courses having 104 credits. In last 3 years, our students are placed successfully in ARS, banks, and other agriculture universities. Our students got a lot of recognition and awards in last year. Dr. Sayanti Guha Majumdar got the Jawaharlal Nehru Award. Dr. Bulbul Ahmed got the Best Poster Award. Bulbul Ahmed also got Young Scientist Award. Parnita Das received Nehru Memorial Gold Medal for Best Student. Nitish Kumar Sharma received Outstanding Research Scholar Award. Our student also participated in various sports organized in IRI and received number of medals and awards. We have faculty of 22, out of which there are 14 research guides. Our faculty also recognized and got awards in different areas in last 3 years, out of which 1 NAS fellow, 2 NAS associate, 2 NASC member, 1 Netaji Subhas fellow, and other awards are there. Dr. Neeraj Budla-Koti received IRI Merit Medal. Apart from this, our faculty has been recognized for presenting best papers in various conferences and seminars. In last 3 years, faculty published 64 papers in various national and international journals, having cumulative NAS score more than 550. Out of this, 28 publications having NAS score more than 10. Apart from this, last year, our faculty got copyrights for 6 products. Faculty organized 10 training programs in which a total of 866 participants were trained. I'll end my presentation with a quote from Duri Duckworth. The one thing that no one can ever take away from you is your education. With this, I thank you all and congratulate each of the degree recipient and their mentors. Thank you very much.
Thank you, Professor Anil Rai, for your nice presentation. Now we will move for the last presentation of the day, and Professor Alka Arora will make presentation on behalf of computer science.
Namaskar, respected Chairman, Director IRI, Dean and Joint Director Education IRI, distinguished members and students present in the session. I am going to present the significant achievements in the discipline of computer application. In the 60th convocation, 11 students will be getting their degrees. In this, 4 are PhD and 7 MSc students. As PhD work is concerned, all students have worked in the broader theme of artificial intelligence, machine learning, and semantic analysis. Kamalika Nath worked under the guidance of Dr. Rajni Jain. She has worked in the area of nature-inspired algorithm for optimization of crop plan, motivation of work. Agriculture crop planning makes a significant contribution to increase production, income, and quality of life in the farming community. Given the limitation of resources, it becomes more important to go for crop planning with the help of optimization techniques like genetic algorithm. The study has been taken up with the objectives to develop single and multi-objective framework for optimum crop plan using nature-inspired algorithms and to develop the IT solution for the same. Under first and second objectives, different nature-inspired algorithms, namely genetic algorithm, differential evolution algorithm, along with its variants, have been studied and crop plan were derived using these algorithms. In the third objective, multi-objective differential evolution combined with Pareto optimality criteria and dominance filtering was used to solve complex multivariate problems and optimal crop plans were derived. Various single objective DE variants were compared and best crop plan was chosen. DE performed superior as compared to other algorithms. Performance of different multi-objective optimization algorithms were compared and mode was found to be perform better than the other optimization techniques. A user-friendly web interface system has been developed to generate optimal crop plans for both single and multi-objective functions using DE algorithms. These are some of the screenshots of the developed system. The approach has been validated using real dataset from the Punjab. DE-based methods outperformed other approaches for crop plants with single and multi-objective functions. A set of similarly good solutions were generated using mode. User can choose the solution considering parameters like water availability, number of workers, equipment availability, capital available, land area, etc. The web-based interface has been developed to solve multi-objective and single-objective optimization problems. These are some of the publications. Muhammad Ashraful Haq has carried out the work on image-based diagnosis of disease severity stages in maize using deep convolution neural networks under the chairmanship of Dr. Sudeep. Motivation. Deep learning-based techniques have shown significant achievements in the field of image-based disease identification of crops by automatically extracting the features from the images. Maize is an important crop, therefore in this thesis, objectives were formulated for application of deep convolution neural network model for diagnosis of disease severity stages in maize along with development of an IT interface for the same. Around 5,000 images of maize crop was collected from the experimental fields of IIMR Ludhiana. Two deep convolution neural networks models, Inception V3 gap, improved Inception ResNet have been trained for disease identification of three important diseases, MLB, TLB and BLSB. Inception V3 gap model achieved the testing accuracy of more than 95% for disease identification. Improved Inception ResNet performed for identification of severity stage of MLB disease with testing accuracy of 92%. Along with deep learning based model development, an AI enabled mobile application severity checker has been developed by integrating the developed model for facilitating the automated identification of disease and their severity stages. Some of the publications are mentioned. Shabana Begum has worked under the guidance of Dr. Rajni Jain on development of particle swarm optimization based model for crop planning. Motivation. Due to severe changes in climatic situation and the continued depletion of available resources, crop planning at a broad scale with optimization of resources is required. Considering this, following objectives were framed: to study the PSO-based optimization algorithm for crop planning, to design and develop the PSO-based model for crop planning and validation of the model. PSO algorithm has been applied on a single objective as well as on multi-objective crop planning approach. Results obtained from PSO model were compared with linear programming and genetic algorithm. The developed PSO model is validated with data from two regions, Bundelkhand and Haryana. Sensitivity analysis done with respect to some percentage of changes in water availability and working capital, which denote the increase of the water and working capital at a certain level will enhance the net returns. A multi-objective approach was designed using multi-objective PSO algorithm using the crowding distance mechanism to obtain the more optimal solution. Along with model development for crop planning, an online software has been developed using R-Shiny that will be useful for researchers in the area of crop planning. These are the publications from the thesis. Sanchita Naha has worked under the chairperson Dr. Sudeep on the topic Ontology-Driven Context-Aware Recommender System for Maize Cultivation. Motivation. Expert to farmers ratio is high, which makes it difficult for experts to reach out to individual farmers. Also, their suggestions are similar for all the farmers in a particular location. However, farmers in the same village or in the same district do not possess the same resources. Therefore, different contextual conditions of individual farmers restrict them to adopt standard recommendations regarding crop cultivation. To effectively cater information to farmers, it is of prime importance to understand their exact requirements and the factors that determine their adoption behavior. Considering this, these objectives were framed. A new ontology-driven context-aware recommendation algorithm have been proposed for suggesting management practices to maize farmers, assisting them from land preparation to harvesting. Relevant context parameters for 8 management practices have been identified through direct interview of farmers using questionnaires. The algorithm has been tested with data of farmers' preferences. These are the screenshots of the mobile application that has been developed. Salient findings. Along with model recommender system algorithm for stage-wise recommendation of cultivation practices in maize crop, a mobile application has been developed to facilitate the same. These are the publications from the thesis. Now coming to the MSc work, which is done under the broader theme of mobile-based DSS development along with application of AI and ML techniques. Subhashish Sarkar has carried out work under the chairmanship of Dr. Mukesh. He has worked on development of mobile-based DSS for nutrition and health assessment using anthropometry techniques. Motivation. Several DSS has been developed in different agricultural activities, but there is no such system available for mapping nutritional status using anthropometric data. The objectives: to design and develop mobile-based DSS for anthropometric measurements related to nutrition and to test the developed system with sample data. The NutriGuide mobile application has been developed using Android Studio Integrated Development Environment, Java and XML. REST APIs has been used with integration with MySQL database. These are the screenshots of the mobile application development. NutriGuide application assists in assessing the physical fitness and nutritional state by calculating different indices, as well as provide a quick note on the value of different types of foods for different age groups. Bharat Kumar N has worked under the chairperson Dr. Sameer Farooqui on the topic Development of Mobile Application for Management of Cole Crops. Motivation: insect pest largely hampers the growth of cole crops and decreases production and productivity. Literature are available on the cultivation practices, disease and pest management in cole crops. Therefore, need was felt to develop a mobile application for dissemination of information to farmers. These are the objectives. Methodology: the cole application, mobile application was created using mobile application development framework. Significant findings: the cole app mobile application provides information of the major cole crops such as cabbage, cauliflower, broccoli, Brussels sprout, kale, etc. System contains information on crops for parameters like cultivation practices, varieties, insect pest diseases, physiological disorder and weeds. Apurva BM has done work under the chairperson Dr. S. Bilal. She has worked on mobile-based management of grape cultivation in subtropic region. Motivation: in order to disseminate the information on various aspects of grapes, a mobile application has been developed with the objectives to design and develop database for growing grapes in subtropics of India, to develop a mobile app for management of grape cultivation in subtropic regions. The mobile application grape culture has been developed using mobile development technologies. This app provides detailed information on vineyard management practice like spacing, planting, training, pruning, fertilization and irrigation, etc. Apps gives information on pest and disease along with management practice and recommends suitable pesticides for particular pest and disease. This application suggests the number of vines that can be accommodated in a given spacing. Pratiksha Shubha has worked under the chairperson Dr. S.N. Islam. She has worked on mobile-based app for integrated farming system. Motivation: integration of multiple enterprises such as livestock, poultry, fisheries, mushroom production, beekeeping, along with traditional crop is needed for a better income and sustainable farming. Multiple IFS models have been developed and those need to be disseminated in an effective manner. Therefore, these objectives were framed. This DSS has been developed using Android Studio, Java, XML and SQLite is used for the database management. These are some of the screenshots showing different IFS models. The system provides advisories for farmers to select the best IFS model. At the same time, it helps the farmers to know the possible profit from the selected model. Lala Saab Momin has worked under the chairperson Dr. S.N. Islam. He has worked on development of web-based application for predicting market price of agricultural commodities using machine learning techniques. Motivation: price forecasting is very important as fluctuating market price of agricultural commodities makes significant negative impact on the socio-economic status of farmers. Machine learning techniques are providing significant results as forecasting is concerned. Therefore, these objectives were framed. HTML, CSS, JavaScript, Python programming language, Django framework, and MS SQL was used for the programming purpose. The agriculture commodity market data was collected from the Ag Market Net. ARIMA model along with the machine learning models, ANN, support vector regression and random forest regression were implemented for the prediction purpose. Web application has been developed. This application was tested with the data collected from Ghaziabad market for rice crop and market price were predicted. Random forest regression performed better in this case. Mobile-based DSS, ISRI, TSW has been developed for prediction of market price data. Bhavesh Kumar Chaubasiya has worked under the chairperson Dr. Anu Sharma on the problem development of deep learning based classification model and web-based application for prediction of LNC RNA in crops. Motivation. LNC RNA are important in crops. Less work has been reported with respect to LNC RNA prediction using deep learning approaches in crops. Therefore, these objectives were considered. Python programming was used for the feature extraction from LNC RNA sequences. CNN-based binary classification model was developed by using Python, TensorFlow, and Keras libraries. The accuracy of model is more than 93%. Web application PLNC predictor uses deep learning based binary classification model. By using this, user can classify LNC RNA from cRNA. It predicts given sequence that whether it is LNC RNA or cRNA. The user can directly predict the type of FASTA sequence by pasting it in the input box of the web application. Sushma K has worked under the chairperson Dr. Sangeeta Ahuja. She has worked on the problem development of mobile app for cultivation of quinoa. Motivation. Quinoa is an important citrus fruit with lot of nutritional values. Therefore, a mobile application has been developed to effectively disseminate the information. The following objectives has been planned: to design and develop database for quinoa cultivation in India and to develop a mobile app for management of quinoa cultivation. These are some of the screenshots. This application provides information about the cultivation practices, varieties, insect pests, disease, health benefits, and computational parameters of quinoa. The application suggests the number of plant population that can be accommodated in a given spacing. Coming to awards and recognition, Nehru Memorial Award by ICAR-ISRI 2021 has been conferred to Mr. Tamal Kundu, MSc Computer Application student. Dr. K.K. Chaturvedi has been recognized for his services as valuable reviewer for International Conference Midas 2021. At the same time, the Best Publication Award for the manuscript in which he is a co-author. The total student passed out, including the 60th convocation in the MSc and PhD are 156 and 7 respectively. There are 16 faculty members and 13 research guides in this discipline. 37 courses offered for MSc and PhD semester system 2021, which covers total 92 credits, including theory and practical. Apart from the regular teaching, senior certificate course on statistical computing is organized at ICAR-ISRI. 3 of our staff, Ms. Sapna Nigam, Dr. Somen Pal, and myself participated in international conferences and symposium in virtual mode. Faculty members were involved in the organization of workshops and webinars. Some workshops were organized jointly with SAUs and Ataris. As faculty members from this discipline are involved in many e-governance applications developed for NARS, sensitization and capacity building programs were organized mainly for e-office, academic management system, ARMS, and Agri-Diksha, benefiting more than 5,000 personnel. Faculty members have published 14 papers in the academic year 2021 in various journals. 10 papers out of 14 are published in NARS rating above 6. Thank you.
Yeah, thank you very much, Professor Alka Arora. So we have completed all the presentation. Now I request Chairman Sir, please your kind remarks and comment.
Oh, thank you, sir. Thank you, Honorable Director of the Institute, Dean Dr. Rashmi Agrawal ji, Convener Dr. Datta, and all 5 professors, Professor Alka Singh, Professor Alka Arora, Professor Sinni, Professor Padaria. My congratulations for presenting very nicely the achievements of the social sciences. Professor Alka Singh is my senior at Pantanagar. He was senior, and my regards to all of you. I have not particular observation, but I have few submission and suggestions from my own. And this is the last group which presented. And one thing I would like to submit and remind that when in 2020, in China, the commission reported that no, now we only teach there are 4 factors of production: land, labor, capital, and management. But in 2021 commission reported, no, now there is another 5th factor, and that is the data science. So I am happy that now the social scientists are having another 5th factor of production, and that is the data science and our data. And it has been proven also, because when we compare Uber or Ola, they don't have anything, only data. They are having data and they are competing in the market. So therefore, it imposes challenges as well as the opportunity for the social scientist as a whole. And I'm very happy that all these divisions are there and they can work together to bring something very fruitful. Now I come to agricultural economics. It is beautiful, very good that the apex institution and the Department of Agricultural Economics is covering almost all sectors, from technology to market, innovation, and other things. My only submission is that when we are talking about IL seeds or pulses, and we are recommending that, okay, the policy initiative should be there. The technology mission was established, then what are the regions, what are the factors which are responsible for not promoting IL seed or pulses? That we have to find out somewhere specifically, so that we can submit to the government. No, these are the areas where the government should come up, because when we see the current budget, this total budget is nothing. It is on the total, it is diversification of agriculture. And it also indicates that the new technology, I can say the Industrial Revolution 4 should be there in agricultural sector. So this is one thing. The second thing I wish to submit with my senior Professor Alkaji. One area is very important. I could not find, and I think we should work. That is the subsidy. I know that there are works done, but the current situation indicators, if you see all 3 F subsidy, food, fertilizer subsidy, food subsidy, and fuel subsidy are the major part of the total subsidy. And even when we see the budget outlay, 2,70,000 crores are allocated for MSP, and what is the allocation of Department of Agriculture as a whole? So the inefficiency, the government is moving in that direction, and therefore the Agricultural Economics Department, we should concentrate on this sector also, which is untouched, which I saw in the achievements. So this is one thing. Definitely to Dr. Padaria ji, I was going through the achievements by the students, and also I suggested then when we are comparing some novel initiative like Sansad Adarsh Gram Yojana, that kind of thing. And if we are not comparing the economic impact, what was before and what was after, the student submitted that no, there is no data. So we should not hesitate to report to the government or report that, okay, if the huge investment is going on in such type of a scheme, and if there is no data before intervention and after intervention, it is a kind of failure, because it is the taxpayer money which is used for this kind of initiative, this kind of investment, or this kind of plans. So I think since both departments are there together, extension and economics, so this kind of intervention should be suggested to the students that no, this should be there. As a whole, the data science, whether it is statistics, whether it is bioinformatics or computer application, my suggestion, my submission is that nowadays each and every person of the country is confident that if in a newspaper says that there will be a rainfall on Wednesday, we all believe that there will be a rainfall. It means, it indicates that in weather parameters, we have become very much confident. The accuracy is 100%. Now that kind of accuracy, we are not able to find till today, whether it is the forecasting of yield, whether it is the forecasting of prices. So I think when we are, may I know that when Sarima X or Garch X is used, in which commodity or which product it was used? I could not find in the text. That's why. When the student use Sarima X or Garch X.
Yes, sir.
So in which crop it was used?
Sir, I'm not sure. Dr. Yasin, you are here?
ठीक है, जिसमें वो भी डाला है क्या इन लोग को, बीटरूट भी डाला है?
Okay, no problem, Professor Sini. Just what I wanted to convey that we have Arima, Sarima, Arch, Garch. We are using ANN. Now we are using deep learning, machine learning, and so on. So can we do something which will increase the accuracy of the forecasting? Whether it is price forecast or yield forecast, it is very good to learn that now the department is doing, and that will help in reducing the crop cutting experiment. That is a very good thing. Now in the last, in most of the MSc study, I could find that it is the development of the mobile app, and that is very good. But how much those apps are practically available, commercially available? So that is a question, and I think that should be carried out. Even the extension department can carry and can do a study that, okay, these different apps developed by the Department of Computer Application, how it could reach to the farmers, and what are kind of supports are required. Unless until it reaches to the farmers, it doesn't matter. It has no meaning. So I congratulate all the degree recipients. Once again, I congratulate all the professors, 5 professors who presented the achievements. And I am really grateful and thankful to Honorable Dr. A.K. Singh sir, who provided me this opportunity. And for last 3 days, I am learning. I am very much benefited by the proceedings, by attending all these proceedings, and really it is a wonderful experience. I sincerely thank to Dr. A.K. Singh sir, Dr. Rashmi Agrawal ji, and the whole team. Thank you very much.
Thank you, Raksham. Thank you very much, and we are indeed grateful to you for being with us for 3 full days. It's not easy for a professor in BHU with so much of responsibility for teaching, that you have spent 3 days completely with us, and your inputs have been very well received, as a matter of fact, and we will try to adopt those recommendations in our research program. Thank you so much.
Sir.
Yeah.
Sir, that Sarima X was for rainfall, and the other one was for price index, which sir was asking. The student is here, sir. So if you want to ask more questions.
No, it was not. In fact, I worked with Dr. Rajnija and Dr. Paul for price forecasting in a network project. We did some study. What I mean to say that when we have new technologies, different whether you have applied all those things, whether deep learning or machine learning and so on, So by incorporating more variables and by using these technologies, now my submission is, can we go for some more accurate prediction that will help the farmer, policymakers, and every stakeholders. That is my submission.
Sure, sir. We'll take it.
Okay, sir.
Thank you. So, Dr. Datta, before you go to say final words of thanks, we have some good news to announce, and I'm sure the students are waiting, although number has gone down to 320. And I'm also happy to see that Dr. Rakesh is there. One thing, I must tell you that the names which have been finally identified for these medals is almost a kind of unanimous decision that happened among the members of jury, although they had only 30 marks in their hand. But by and large, when those marks have been added to the academic scores, the brilliant ones have been, all are brilliant. I'm not saying they are not, they are all brilliant, but those who are identified in presentations, by and large, there has been a good coherence and coincidence in that they are together. So I'm pleased to, announce the results for MSc. And are students there? Can you say a cheers? Okay.
Yes.
So,
Great,
good. So first,
yes, sir.
Great, good. First, let's come to MSc based on the marks obtained. That is OGPA, research paper published, special achievements, awards, co-curricular activities, which was for 70 marks, and thesis presentation and discussion 30%. That was 70%, this is 30%. Following MSc students have been selected for the best student of the year 2021 and NABARD Professor V.L. Chopra Gold Medal and IRI Merit Medals. So the best student of the year and NABARD Professor V.L. Chopra Gold Medal Award goes to Miss Devrati Mondal from Biochemistry. Okay, a big round of applause for her great achievements. And the IRI Merit Medal go to Mr. Shinto Antu, Plant Physiology, Miss Komal G, Entomology, Mr. Uttarayan Das Gupta, Genetics and Plant Breeding, Miss Adiga Amla, Floriculture and Landscape Architecture, Mr. Sarat Kothari, Soil Science and Agricultural Chemistry. Congratulations to each one of them. Hearty congratulations. Now, coming to PhD, of course, the same criteria, the guidelines were same. The following that, the best student of the year and award Professor V.L. Chopra Gold Medal Award goes to Mr. Siddharud Maragal, Vegetable Science from Indian Institute of Horticultural Research. Big clap for him, and also clap for the respective chairpersons for MSc as well as for PhD for guiding the students. I understand the chairperson is also an IRI alumni, and because I just looked at their photographs, so I could recognize he used to do plant breeding courses with me. So that's how I could recognize him. Hearty congratulations to director also of IHR. And the IRI Merit Medal go to first Mr. Kuleshwar Prasad, Plant Pathology, Mr. Ashish Khandelwal, Agricultural Chemicals, Mr. Neeraj Kumar, Genetics and Plant Breeding, Mr. Kaushik Banerjee, Agricultural Physics, Ms. Uma Prajapati, Post-Harvest Technology. So congratulations to all the winners, and also the others who are not in the list, they were 1 to 1 and second to none. So congratulations to them also for being, in the race, and to all the chairpersons. And thank you all very much for being here in large number. Thank you so much. Now, Dr. Datta, over to you.
Okay, sir. Thank you very much, sir. This is a last session. We are at the end. So it is my pleasant duty to propose formal vote of thanks. So at the outset, I sincerely thank Professor Rakesh Singh ji, Head and Professor, Economics Department, BHU, for readily agreeing to our request to chair the session and share his valuable experience. I am sure, sir, your appreciation will encourage our students and faculty, and your critical comments will help us in deciding the future course of action. Thank you very much, sir. I am highly grateful to our honorable and beloved director for guidance, constant support, encouragement, and inspirational scientific inputs. In spite of your busy schedule, sir, and huge professional responsibility, whole day you have been with us and actively participated in the scientific discussion, and you also intervened meaningfully wherever required. This shows your inclination and love for science, and you are really a great source of our inspiration. Thank you very much, sir. Thank you. I am extremely grateful to our Dean Madam for guiding cooperative attitude, immense patience, and useful discussion during the course of organizing this program. And Madam have been actually busy for now also busy due to convocation, but in spite of this her busy schedule, she could go through the publication, that compilation we prepare on significant achievement of students' research. This shows her commitment, and that really improved the quality of that compilation. Thank you very much, Madam. I feel immense pleasure to convey my heartfelt thanks to Dr. K.M. Manjaya, Associate Dean. He was always there in all my needs and helped his best whenever I sought. Now coming to the professors, I am more than indebted to the professors, to all the professors, those who have made presentation, for their excellent presentation as well as maintaining the time. In fact, sir, some of the presentation I returned back that you have taken more time like this, but they all rose to the occasion and really helped me, and they reduced the time. And that's why, sir, we could complete almost within stipulated time all the presentation. I thank profusely to our students, because you are the focal point. Your work actually has been presented by the professor. So without your work, actually nothing could have moved. So thank you very much. And I thank Dr. R.R. Burman and Dr. Amarinder Jha for efficiently managing online system. Thank you very much. And there is rightly said, there is no substitute of experience. Yes, I am indebted to all the senior people like Dr. H.S. Gupta, former director, H.S. Gaur, former dean, Dr. Lodha, and many others virtually joining this program. Sir, your presence indeed inspired us one and all. Thank you very much, sir. Finally, I pray to Almighty and hope that this next convocation will be on physical mode. Thank you. And I am really grateful I'm sorry I missed some. I am really grateful to heads, scientists, staffs for joining the program and made this program grand success. And finally, I will conclude with a quote of Swamiji. This is for student only, mainly for those who are going to get degree. Once Swami Vivekananda actually differentiate between the education and literacy by putting one example. He says, if one atom of oxygen combines with 2 atoms of hydrogen, it forms scientific fact. If we know this fact, we become literate about that. That is what we are after getting so many degrees and diplomas. We will be educated if we realize that we can prepare water, we have water, and this water should equally be shared by all thirsty people. That is education. So with this message for my student friend, I will stop here. Thank you. Thank you very much, sir.